Hello and welcome. As usual, I'm Corey Campbell. I hope everyone's having a great week so far. The last time I streamed was on Sunday, so it's been whatever, 24, 36 hours since my last stream, so not much can happen. But yeah, I've been having a pretty uneventful week so far, in a good way. I pretty much just went to the gym yesterday, and then, got some work done. That's basically it. Oh, and actually today, so, I was, again, as usual, I do my fitness streams, or workout streams on Tuesday. And, I went to the private gym studio, and, not gonna lie, similar to last week, I just, was not in the best mood to stream my workout. I just kind of wanted to, be alone with my own thoughts as I was working out. Trying to focus on form, trying to, also, work out some things in my brain at the same time as working out. And so, yes, I decided not to stream my workout stream, but working out felt good. And I'm glad that I went, and I'm also glad that I'm still maintaining somewhat of a normal routine despite this minor health issue that appears to be quite ongoing and chronic, and I don't really know how long it's going to last. Again, still in the process of trying to track down a surgeon that actually does the minor operation that I need. So far, all the surgical clinics I've contacted, I pretty much get turned away, saying, we don't do that here. So the search continues. But I'm sure I will find something eventually. And on a somewhat different note, I might be changing jobs, maybe, tentatively. It might... So I currently, in addition to pursuing my PhD, I also have a part-time job at a biotech startup. I've said many times before, basically a scientific instrumentation company. And I've applied for a new position at a different company, much larger company, basically a fully established, kind of international company that has head offices, in multiple locations throughout the world. Without giving, too much away, but if in fact I get this position, it might become my job to read scientific or medical literature and put it together in a sensible fashion. I can't really get more specific than that, other than it will be my job to read and write about scientific, or more specifically, medical research. So potentially excited about that. It's actually a fairly lengthy interview or screening process. There was the initial interview that was 2 weeks ago, maybe 3 at this point, that I thought went well. And well, I guess I'm pretty certain it went well, because there's technically 3 stages to the full, interview screening process. Stage 1 is interview number 1. Stage 2 is they actually give you a take-home test to basically test out your writing ability. And, can you actually do research or read medical research and actually, put together a coherent document. So I got past the first interview, and they sent me the take-home test, which I completed and submitted exactly a week ago. And then I got a response today, inviting me to the third, what should be the last stage, is final interview. That's going to take place sometime this week. Oh, and anosmia. And I apologize if I mispronounced. I know you told me the correct pronunciation, but anosmia, I think, was the pronunciation. If not, correct me. Thank you so much for passing by. I hope you're doing well. I'm just giving a sort of an update on myself. where it's like, yeah, I pretty much got through the first 2 stages of the screening process. Now I'm in the 3rd one. The last interview will take place sometime this week. And then I will hear back whether or not I got the job. Anosmia. Okay. I'm gonna do my best to commit that to memory. I do want to get it right. So thank you very much for being patient with me. And, you know, it's potentially exciting, as in, if I get this job, and, you know, like I said, this job would mean that I literally get paid to read medical research and, you know, summarize things and put it together in a coherent, hopefully coherent fashion. So that would be cool. Also an excellent question. From what I understand, the pay will be substantial. About to sneeze, I think. Alarm. Not false alarm, just delayed sneeze. So, thank you very much. So as of right now, in my current part-time job, I am paid, technically, $30 an hour. Which for someone who's a PhD student, that's not bad. Or that's as a part-time job, that's pretty well above what the current minimum wage is here in Quebec. And it technically pays better than me teaching at the university. Teaching at the university, you're paid around, like, well, not a full tenure professor, since me being a teaching assistant, you are paid around like $25, $26 an hour, which is why I went with a part-time job with this biotech startup, because it pays better, and also, like, it actually uses my expertise. There are not very many people, at least locally, you know, in North America, that have the type of expertise that I have, that this company basically, you know, uses. Yeah, I'm definitely not complaining. And $12.50, I don't know if that's Canadian or American. I think that makes a difference. And I know definitely if you're American, like, I think the minimum wage, like, hasn't changed in decades. And yes, another excellent question. I would be doing this pretty much from home. Like, there's an office space here, but, like, I would have virtually no use for it. Like 95% of my work would be home-based, or like virtual home-based, you know, writing and virtual meetings to that extent. And so $12.50, I think that translates to, like, I guess $15 Canadian. Let's do 1 second. Let's do the conversion. I'm curious. $16, yeah. And I think like the Quebec minimum wage is like I think around 12, 11 or something like that. Oh no, 14. Okay. So yeah, technically speaking, anosmia, who are being paid above Quebec minimum wage. Not that Quebec minimum wage is all that great. Oh, and Avenging Pineapple, thank you so much for passing by. Much appreciated. I can't believe anyone would make it. Well, we're gonna get into that. Cover a little bit of the fraud. And, well, that's the thing. You know, there's the issue, I would say, well, we'll get into it, but bad incentives. And, you know, our current institutions of science, I would say, you know, don't incentivize. I think I understand the joke that you're trying to make, but still, I'm going to take it seriously. That, how our scientific institutions run, they don't really, it's not conducive to attracting the type of people that want to do, good science, that want to do good science for the right reasons. But we're going to start that in a bit, I guess, just to wrap up. This is potentially an exciting job opportunity. And to answer also your question, Anosmia, it would be a substantial increase in pay or salary. I would essentially be paid double, if not technically more than double. So again, potentially exciting. But that's not for sure. The last and final interview takes place later this week. I'll obviously keep people updated. And I might have some difficult decisions to make if I actually get this job. Because to answer your question, Anosmia, it would technically be a full-time position. It would not be part-time. And that's why it's like more than double. I would have to, rebalance some things. I might have to pursue the PhD part-time. Maybe put it on hold, actually. I've sort of, I've not committed myself to anything. Yeah, it's pretty much the exact same thing here. 37 to 40 hours. Or, on paper, okay, 40 hours. But then, as I'm sure most people know, many corporations don't necessarily honor that. Or rather, they will extract and squeeze every ounce of labor out of you as much as they can. Which is why I generally don't like working for corporations. Because I'm not the best. I will fully admit I'm not the best team player. Once my 37 or 40 hours are done, I pretty much say, like, that's it. You got what you paid me for, and anything else, you need to start paying more. But either way, I'm getting way ahead of myself. But this opportunity presents some interesting things to me. In terms of, it allows me to really test my ability to, read scientific or medical literature and write about it. And, I'm assuming if a, large company sees that, I'm able to read, research, and write about it, and they're happy with my work, I'm hoping that, instills confidence, that, people can trust my ability to read and translate science. Not all science. But just to, pick up a concept quickly, research about it, and talk about it. Because the take-home test that they gave me, it's on a specific topic. It's on a specific medical topic. I can't say what it is, but it's technically not a medical topic that I had any previous knowledge about. Totally brand new. Like, the test is basically like, here's your medical topic, write about this. Here's your disease and condition, research it, write about it. Here's sort of the theme that you should be writing about. And I did it. Or rather, I did my best. And it seems like, at the very least, they were satisfied. And I think that's why they asked me for, the final interview. Or, put me through to the final round. So even that, in and of itself, sort of, at least, gives me a little bit of a confidence boost. Because I know, I know that I'm competing with other people that have, prior experience in this field. Like, technically speaking, you look at the application, like, I'm technically not qualified. They were They were asking for literally at least 5 years of experience in this field. I have 0. And so, again, if I'm able to get through and actually get the position on 0 years of experience, well then, yes, that's a little bit of a confidence boost for myself. Obviously, it's not the only thing that matters, but still, it'd be nice. What is the housing market like there? I have a couple of friends that live in Vancouver, and they say so, well, at least I'm certainly no expert. I only mostly, I read some headlines and some articles here and there. But, I know some friends and colleagues that are in the housing market, and it varies from province to province. But, generally speaking, overall, across the country, it's not good, But I hear some of my friends talk that it's, getting better, or that, it's supposed to get better in the near term. But that remains to be seen. What I do know is that, I have a couple of friends or colleagues that are they're actively searching for a house. Okay. And these are well-educated people with stable salary jobs, where it's I have one friend, they have graduated with their PhD. They have done a postdoc. They are working full-time, salaried position for this company. And it's not like it's a bad salary. I know exactly what their salary is, because I was actually their witness to their contract signing. So I actually know how much money they're making. And yet despite that, they still can't find a home that's in their price range. Or they basically can't find an affordable home for them. And this is again, a highly educated person, more educated than me. They actually graduated their PhD. They did extra, postgraduate education, a postdoc. And still not able to really buy a house just yet. And is rent affordable? So, I mean, it depends on what your expectations are. If you're looking for, a small place for one person, and you don't have a requirement for 3 rooms, then I would say yes, rent is affordable. But that's all relative. I would say, generally speaking, Quebec, in comparison to the rest of Canada, is generally speaking more affordable. But even here, rents are going up, and it seems to be, trending in the wrong direction. But overall, as of now, for small spaces, basically kind of student-like spaces, they are still sort of in the ballpark of affordable. But I don't really know how long it's going to last. Alright, so we're just going to get right into it. Actually cover the topics in this stream title. This story came out, I think, a week ago, a little over a week ago, the 21st of July, about basically what looks like. So I haven't read this article, so this is new information. Whatever, the contents of the article are new to me, but not the headline, not the knowledge that there appears to have been fraud committed in Alzheimer's research. But yeah, it seems like there was essentially, doctored data or fabricated data, or data that was essentially photoshopped. And it appears like it has had, widespread ramifications throughout, the Alzheimer's field. Oh wow, well, it's totally not necessary for you to donate. It's super appreciative, and I'm grateful, but I don't want to. Don't feel obligated, because you're not obligated. But if you want to donate, I'm super grateful for that, and I can't thank you enough. So this is published in Science, okay, but this is not technically it's not a scientific article, right? This is not from a scientific publication. This is more like a sort of an editorial, or a journalistic expose covering this fraud. So, in August 2021, Matthew Schrag, a neuroscientist and physician at Vanderbilt University, got a call that would plunge him into a maelstrom of possible scientific misconduct. A colleague wanted to connect him with an attorney investigating an experimental drug for Alzheimer's disease called Simufilm. That's, as usual, drug names, always tricky. Oh wow, Anosmia, thank you so much.
$3.
You're super generous, and I can't thank you enough. Again, super grateful. That's very kind of you. And you know, I'm gonna do my best to hopefully continue to bring you good content and live up to that. So, yeah, the drug developers, Cassava Sciences, claimed it improved cognition, partly by repairing a protein that can block sticky brain deposits of the protein amyloid beta. And so it might be useful to maybe give a little bit of a brief primer on Alzheimer's. Even though I think, virtually everyone is familiar with Alzheimer's, either is familiar or is somehow indirectly affected. I think just about everyone at this point either knows someone or cares about someone or has someone in their life that will be affected, either directly or indirectly by Alzheimer's. Just because, yeah, it's unfortunately so prevalent. And I'm certainly not an Alzheimer's disease expert, but I certainly, know a decent amount about it, and I've taken advanced neuroscience courses, and we actually cover Alzheimer's in it. And actually, as part of this advanced neuroscience course, we had a guest lecturer that spoke specifically about Alzheimer's, because their area of specialty was Alzheimer's. And generally, in a nutshell, Alzheimer's is essentially kind of certain regions of your brain, largely the hippocampus. Okay, and this is one of the reasons. This is pretty much the reason why Alzheimer's is associated, strongly associated, with memory issues or straight memory loss. It's because, you have this essentially degradation of the hippocampus. And the hippocampus is essentially where memories, long-term memories are stored. It is essentially, in many respects, like the hard drive of your brain. And Alzheimer's, specifically affects that. And if you look at, the brains of those affected by Alzheimer's, and you actually, look at their hippocampus, like you see that like it's like actively being kind of like eaten away, or like starts to kind of like resemble like Swiss cheese, of like actual like holes are like being poked through these areas of the brain. Obviously it's not not the result of something external. There's like an actual like biological, physiological process that's happening, that it's resulting in this area of the brain sort of actively kind of wasting away. And the overall hypothesis, as far as I know, is that it's a protein misfolding disease, as in proteins. Okay, they are large macromolecular structures, and they adopt certain intricate 3D structures. Like they need to be folded in a certain way. And then once they start unfolding, they start losing basically their function, because their particular 3D structure is what allows them to carry out their function. And so like, again, if these proteins start misfolding, there's kind of like a domino. There appears to be kind of a domino effect, as in like if one protein in an area misfolds in this particular sort of like disease process, it seems like there's like a cascading or domino effect, where if just one protein starts to misfold, well, then it results in like the protein nearby starting to misfold, and then on we go, in a progressive fashion. This is why it seemed, our understanding of Alzheimer's is that it's a progressive disease. It's essentially just going to get worse over time. Do you think that microplastics in our blood has anything to do with rapid increases in Alzheimer's rates? They found microplastics in the brains of unborn babies a few years ago. So you are asking a fantastic question. And the research, as far as I can tell, on microplastics, like, is in its infancy. Like, we are only just beginning to kind of tease out the effects of these microplastics accumulating in our bodies. Like, we pretty much know that now, as in, like, pretty much every human being on the planet now, like, some small portion of them is, like, made of plastic, 0.01%, 0.001%, but, like, we're consuming plastic, it's accumulating in our bodies, and it just, accumulates over time. And I've touched on it a little bit. 1 second. Or it's like, yeah, I brought up some of this research before, where it's like, it's pretty much has contaminated or is present at every level in our food chain. From fish to animal agriculture, to our plant foods, etc., etc. But what we do seem to be finding out is that, generally speaking, the animals, particularly seafood, they very often have much higher concentrations of microplastics, and that if you want to minimize the amount of microplastics that you are consuming, you should probably at the very least switch to a plant-based diet, because the lowest concentrations of microplastics, appear to be in plants, which makes sense because plants don't physically eat anything, and so it's hard for something to bioaccumulate. And like, again, sort of what I was saying, like, we're only really sort of beginning to understand some of the biological or physiological effects of these microplastics, or microfibers, right? Like, there's just all kinds of micro stuff now in our environments. This is published in 2020, refers to immunotoxicity, etc., of, nano and microplastics. And unfortunately, I have very little doubt that more research will come out showing that, the effects are almost certainly not good. Of the research that we have now, it doesn't seem like, it's good to have these plastics accumulating in our bodies. It doesn't seem like the worst. But, if we ever find a way to, like, somehow rid our bodies of these microplastics, that would be probably a good thing. I know that the average American consumes 1 to 5 grams of plastic a week, and I think it was about 0.1% of that gets absorbed into our blood. Yeah. And obviously it's going to vary based off of, like, your diet, etc., etc. But, like, yeah, if you are actually concerned about the amount of microplastics that you're eating, probably eating less animals and eating more plants is probably a good decision. Yes, exactly. There appears to be sort of a hormonal or endocrinological effect, where, like, yes, it seems like one of the issues with these microplastics, okay, it's arguably that plastics themselves aren't the issue, as in, like, the plastics alone, them accumulating in our bodies, may not actually be like, the worst thing. But the real sort of damage or negative effects, they are coming from like all of the stuff that is like just hitching a ride on these plastics, on these microplastics, which include chemicals. A lot of them have, effects on our, it looks like actual hormones are sort of hitching a ride on these microplastics, and then we're eating them. And they're affecting our hormones at some level. And so, it's the next, I think, few years of research about this, I think, is going to yield some interesting and probably not good things, unfortunately. But yeah, getting back to Alzheimer's. So, yeah, with that in mind, that little brief primer of Alzheimer's, everyone knows it's a brain condition. And that, it affects memory. It affects memory because the hippocampus of the brain is essentially being slowly degraded over time in Alzheimer's patients. The attorney's clients, two prominent neuroscientists who are also short sellers who profit if the company's stock falls, believed some research related to simufilam may have been fraudulent, according to a petition later filed on their behalf with the US FDA. Bragg, 37, a soft-spoken, nonchalantly rumpled junior professor, had already gained some notoriety by publicly criticizing the controversial FDA approval of the anti-alpha-beta drug Aduhelm. His own research also contradicted some of Casaba's claims. He feared volunteers in ongoing simufilam trials faced risk of side effects with no chance of benefit. So he applied his technical and medical knowledge to interrogate published images about the drug and its underlying science, for which the attorney paid him $18,000. He identified apparently altered or duplicated images in dozens of journal articles. The attorney reported many of the discoveries in the FDA petition, and Schrag sent all of them to the National Institutes of Health, NIH, which had invested tens of millions of dollars in the work. Casaba denies any misconduct. But Schrag's sleuthing drew him into a different episode of possible misconduct, leading to findings that threaten one of the most cited Alzheimer's studies of this century, and numerous related experiments. So, it seems like, this fraud that's been exposed, like, it took place in, like, a landmark study. As in, like, when this study came out, it was like, oh, this is a game changer. And now, like, 15, 16, 17 years later, this comes out saying, oh, maybe this was fraudulent research. And that, the avenue that it took us down might have been the wrong road all along. Maybe we should have stuck to our original road. But we'll go on. The first author of that influential study published in Nature in 2006, and Nature, as I've said, many times before, a very well-respected, highly regarded journal. This probably brings us to that particular study. Yeah, about Alzheimer's. A specified amyloid beta protein assembly in the brain impairs memory. Was an ascending neuroscientist, Sylvain Lesny of the University of Minnesota, Twin Cities. His work underpins a key element of the dominant yet controversial amyloid hypothesis of Alzheimer's, which holds that alpha-beta clumps, known as plaques, in brain tissue are a primary cause of the devastating illness, which affects tens of millions globally. In what looked like a smoking gun for the theory and a lead to possible therapies, Lesny and his colleagues discovered an alpha-beta subtype and seemed to prove it caused dementia in rats. If Schrag's doubts are correct, Lesny's findings were an elaborate mirage. 1 second, I'm just gonna adjust the volume a bit, or the music, background music. Schrag, who had not publicly revealed his role as a whistleblower until this article, avoids the word fraud in his critiques of Lesny's work, and the cassava-related studies, and does not claim to have proved misconduct. That would require access to original, complete, unpublished images, and in some cases, raw numerical data. I focus on what we can see in the published images, and describe them as red flags, not final conclusions. the data should speak for itself. A 6-month investigation by science provided strong support for Schrag's suspicions and raised questions about Lesnay's research. A leading independent image analyst and several top Alzheimer's researchers, including George Perry of the University of Texas, San Antonio, and John Forsyth of the University of California, San Francisco, reviewed most of Schrag's findings at Science's request. They concurred with his overall conclusions, which cast doubt on hundreds of images, including more than 70 in Lesnay's papers. Some look like shockingly blatant examples of image tampering, says Donna Wilcock, an Alzheimer's expert at the University of Kentucky. Bob Marley, hello. Welcome. Thank you so much for passing by. I'm hoping you're still enjoying your time in Croatia. Or maybe you moved on from Croatia already. And, just to sort of give you an example, let's see. All right, this is just, obviously, many scientific publications. Okay. This is just a random one. Well, not random, but, random to you. But, scientific publications, especially in the life sciences or physical sciences, they publish figures, pictures as well, obviously. And, it's kind of we basically go by the honor system, in that, we go based off of trust that, okay, what you're putting when you write it up, when you write up your scientific publication, that is the truth. You are not doing sort of any real editing. You're not using Photoshop. You're not cropping things in a manipulative manner. Okay. It's just, we go by faith that, okay, these pictures that you're showing us, these are undoctored pictures, and that, this represents reality. But there are, quite a few documented cases of scientific publications having pictures that have almost clearly been photoshopped, as in just, some researcher decided to hey, we're gonna put this here in the picture. We're gonna make it look like the results that we want it to look like. And it's only sometimes, very far down the line. In this case, over a decade later in this Alzheimer's research of we find out, oh, maybe there were some, doctored or basically photoshopped images in this research. And even here, it's taken on faith that, what you're showing, that this labeling is true, okay? That this is, because what you're shown here in diagram G, it's literally, this is a heavily cropped image. This is from a much larger image that contains lots of little black bands, and they are just zooming in and, cropping the bands of interest. And it's just going, when this is published, we're going on faith that, all of the stuff that isn't shown, it's not relevant. This is a Western blot. If you actually know what a typical Western blot looks like an actual fully developed one, like the original, doesn't look like this. Let's see if I can find an example. Right. So a Western blot, looks like that. Lots of bands. Okay. And you can, hopefully you can see that on this one, this is a Western blot. Doesn't look like this. You know, you're not seeing the large rectangle and the bunch of bands, dark bands. What they've done here is they've just cropped the bands that, you know, they want to show. The ones that are, you know, whatever, scientifically relevant, you know, for this paper, for this research. Because, you know, we go on faith that, you know, that's what researchers do. They don't have ill intentions. We're not looking to actively hide something or, you know, manipulate the reader. But, you know, ideally, you know, you would show, like, this is technically the entire data set. And they're just showing, like, let's zoom in on one particular band and show that. Like, they're just, like, zooming in so heavily. Oh, back in Germany? Okay. I'm hoping you had a great time in Croatia. So there isn't typically an investigation on these studies. So that's where it gets tricky. Because, like, absolutely, virtually all good scientific research is peer-reviewed. But it's peer-reviewed under this honor system. Where it's like, you know, well, I mean, I've submitted scientific publications to peer review. Like, I've had my work peer reviewed. And it's like, you know, you type up sort of like your final draft, okay? And it includes sort of like the figures that you want to include, zoomed in on the things that you want to include. And the peer reviewers, like, unless they actually, like, notice something like that's obviously doctored or, like, you know, is actually fraudulent, the peer reviewers, like, they go on trust that, okay, like, they're not, the scientists are not trying to hide something. They're not actively engaged in deception. Which, you know, that probably holds true, I'm hoping, for, like, the vast majority of scientists. But, you know, we have clear cases. And, like, the cases that, you know, I'm going over now in this Alzheimer's research, that, you know, there are some bad actors in science. Way too many bad actors, where it's like, for the sake of their career, for the sake of trying to publish something with a high impact, or for the sake of trying to be the first to publication, you know, they will engage in, you know, sort of scientific, you know, misconduct, scientific fraud. Where the line is between misconduct and fraud, I don't know for sure. Look at the no, I have not. I don't think I've even heard of it. But if you have maybe a link send my way that explains it, I'd be more than happy to take a look. So the authors appear to have composed figures by piecing together parts of photos from different experiments, says Elizabeth Bick, a molecular biologist and well-known forensic image consultant. The obtained experimental results might not have been the desired results. And that data might have been changed to better fit a hypothesis. Ooh, that's also a really good question. So, basically, it depends on, you know, the extent of fraud. But in general, there's not really much, quote-unquote, punishment. Other than if it's shown, okay, that your research is fraudulent, like explicitly fraudulent, then it will likely just get pulled from the journal. And, you know, you'll just be notified saying, hey, your research has now been, you know, rejected or pulled from our journal. And that's pretty much it. There's typically not, like, some sort of huge disciplinary action as a result of scientific fraud. Maybe, like, if a university learns that one of their professors has, you know, engaged in, you know, multiple cases of scientific fraud, maybe the university will do something. But in general, there's not really a lot of punishment, except for just the reputation. As in, like, you know, if you get exposed for scientific fraud and, you know, your university lets you go, you will almost certainly have, you know, a pretty difficult time, you know, finding a new position at another university. It's the first big vote on abortion. Kansas citizens are voting on whether to keep the right to... Ooh, okay. I will have to look that up afterwards. Or, like, yeah, if you have a story, send, by all means. Yeah, it's pretty much character suicide. That's probably the more appropriate way to frame it. Where it's like, we're hoping that one's desire to keep a reputation in science is what, pushes them to do good science. But I think we have great examples that doesn't actually happen. Early this year, Schrag raised his doubts with NIH and journals, including Nature. Including Nature last week, have published expressions of concern about papers by Lesney. Schrag's work, done independently of Vanderbilt and its medical center, implies millions of federal dollars may have been misspent on the research, and much more on related efforts. Some Alzheimer's experts now suspect Lesney's studies have misdirected Alzheimer's research for 16 years. The immediate obvious damage is wasted NIH funding and wasted thinking in the field because people are using these results as a starting point for their own experiments, says Stanford University neuroscientist Thomas Sudoff, a Nobel laureate and expert on Alzheimer's and related conditions. Where it's like, yeah, it seems like maybe one of the biggest consequences, of this fraudulent research is that it might have led other researchers towards a dead end, for decades. As in, like, this fraudulent research, well, now sort of known to be either fraudulent or problematic. Like, at the time when it was published and it was seen as true, it was almost like a paradigm shift of like, oh, we thought we understood Alzheimer's before, but now with this new landmark paper, we have a new and better understanding of Alzheimer's. So then let's take that and run with that. But it seems like maybe, it might have just led lots of researchers towards a dead end. If in fact, this turns out to be, absolutely fraudulent and, no basis in reality. And usually for elections in Kansas, turnout was in the mid-30s, and for this vote, early estimates say 50%. Interesting. Okay. Definitely going to look that up afterwards. How can a person like me who doesn't have any higher level education determine what studies are accurate and who is of high reputation? Is there a database? Excellent question. There's this website here. And here is where I want to be careful in what I say. Well, I'm hoping I'm always attempting to be careful in what I say. But there's the difference between fraudulent research and, research that can be criticized. Or, research that is, maybe highly flawed. The ones that are actually fraudulent, that, is basically bad science. We don't catch them all, but there are websites devoted to, basically catching fraudulent research. It's called the Retraction Watch. It's called Retraction Watch because, typically when research is shown to be fraudulent and it's already been published in an academic journal, it is retracted. As in, if the evidence is put forth that, hey, this paper is fraudulent science, and then, they go through the review and they conclude themselves that, yes, we agree, it's fraudulent science, then, that journal will retract that paper saying, hey, this is no longer basically part of science. And so that's what this website is devoted to. And so this is fraudulent research. And to answer your question of like, how can you determine what's accurate? This is where a little bit of faith has to come into play. Unfortunately, even for myself, I have to have faith when I read certain papers, papers that are outside my field, that are way outside my field, that, you know, I don't have the knowledge, the qualifications, or expertise to be able to, like, you know, really understand their methodology or why it is they're doing what they're doing. And so just be careful. Like, basically, unless, like, shown otherwise, you know, I operate under the principle that, you know, if it's published, I will take the science at face value. This is not to say that, you know, I will absolutely treat it as gospel and like the absolute truth. But I try to do my best to say, like, whatever, even if I disagree with its methodology, even if I can, you know, poke holes and criticize a study, but that doesn't mean that that publication is fraudulent or that it's quote unquote debunked. It just means that, you know, it may not be the highest quality of research, and that, you know, there's a grading scale to evidence. So yeah, to answer your question, there are websites like this. This is, I think, one of the most notable ones, if not the most popular. Retraction Watch. If it appears on here, then it's almost certainly to be fraudulent research. But generally speaking, you know, anything that's published in a peer-reviewed academic journal, you know, take it at face value that it's, at the very least, decent research. Oh, and Noda Bud, welcome. Thanks so much for passing by. Hope you're well. Let me just see. Well, I think there's a middle ground there. Of, like, I agree with you, people are not dumb, but I don't know. I'm not sure I'm fully on board that everyone is equally equipped to read academic scientific research. Yeah, you need to have a critical mind and an active goals and methodology. Even if you have a critical mind, that's I guarantee you, a critical mind is not enough to poke holes in methodology in every single scientific paper. I would absolutely put that to the test. And well, yeah. So I'm definitely of you know, Bob Marley's sentiment of, like, you know, many people that I know that have no business talking about vaccines or virology. You know, they appear to have become vaccine or virology experts. Everything the anti-vaxxer said came true. So I'm curious as to what exactly, like, what specific claims are you saying came true? Because I would, that's not my understanding. And are you saying, for example, that they got hydroxychloroquine true? That they got ivermectin true? If you want to clarify, but I'd be very curious to know, like, what specific claims that you think that they got true. Methodology for everything in the first, well, but that holds true for every single study, like, in existence. There is no such thing as research that is not flawed, even from a methodology standpoint. Every single piece of scientific research that is published, even my own, okay? Like, I can look at my own research and poke holes in that and say it's flawed. But if you're gonna come and say, oh, that means my research is fraudulent, well, then I would say you probably don't know what you're talking about. They said we would be taking more than one, then more than two. They said they would try to give them for years. Yeah, okay, but that's, like, as far as I know, even those who are pro-vaccine, they were not making the claim that it's only going to be, like, one dose. Because, like, even first of all, Like when the first mRNA vaccines came out, I think they were already like 2 doses at the bare minimum. Are you seeing the methodology over the Hodgkin's? Proper? The research people point to the most literally gave above recommended doses. Well, so that's pretty much my point. Like, so it seems like almost in your framework, like no study can ever prove a therapy effective. Okay? Like it seems like your bar to prove that any sort of therapy is effective is so high that no drug in existence would ever meet it. Because again, even randomized placebo-controlled trials, they are flawed and they are seen as like the gold standard. Let me see if I can bring it up. Whoopsies. Big. Okay. So this is from, the journal whatever, Neurology. I'm just using this as an example. But I mean, they literally have their own published ratings for how they classify evidence, scientific evidence. And you can see, okay, that they have, class 1, that's the best, class 2, class 3, class 4. And as you can see, class 1 is a randomized controlled clinical trial of the intervention of interest with masked or objective outcome assessment, blah, blah, blah. And they have like all of these other criteria, as in like this right here, this is the highest quality of evidence, okay? And this is the criteria for like the Journal of Neurology. And even this, okay, this is flawed methodology. Like you can criticize any scientific publication that falls under class 1. You can poke holes in its methodology. You can show that it is flawed research, but that does not mean that you can discount it, or that you can't trust its conclusions. And so to rely on this argument of like early COVID research was flawed. Well, yes, even late COVID research, every research is flawed. I would say, Notable, you have to make a better argument than that, other than to just say, well, it's flawed. Well, flawed in what way? Because every single paper in existence is flawed. One second. Zero signs for lockdowns. At the time, I mean, this is a brand new. At the time, that was like a brand new situation that had like never occurred before. Of like a global spreading virus that was like fairly highly transmissible. Like, how would we even run that experiment? Of like, is a national lockdown effective? Well, at some point we have to nationally lock down and try that. Like, how do you, like, based off of your metric, okay, or at least your standard of evidence, we would have never concluded that smoking is bad for you. or that smoking is, a causative agent of cancer. Where was I? Oh, yes. Here. Schrag, who had not publicly revealed his role as a whistleblower under this article, avoids the word fraud in his critiques of Lesnay's work and the Cassava-related studies, and does not claim to have proved misconduct. That would require access to... Well, I already read that. A 6-month investigation by science provided strong support for Schrag's suspicions and raised questions about Lesnay's research. A leading independent image analyst and several top Alzheimer's researchers, including George Perry of the University of Texas, San Antonio, and John Forsyth of the University of California, San Francisco, reviewed most of Schrag's findings at Science's request. They concurred with his overall conclusions, which cast doubt on hundreds of images, including more than 70 in Lesnay's papers. Some look like shockingly blatant examples of image tampering, says Donna Wilcock, Alzheimer's expert at the University of Kentucky. The authors appear to have composed figures by piecing together parts of photos from different experiments, says Elizabeth Bick, a molecular biologist and well-known forensic image consultant. The obtained experimental results might not have been the desired results, and that data might have been changed to better fit a hypothesis. Early this year, Schrag raised his doubts with NIH and journals including Nature, last week, have published expressions of concern about papers by Lesnay, Schrag's work. Yeah, I already read this. Behind. Yes. Oh yeah, Lesnay did not respond to requests for comment. A UMN spokesperson says the university is reviewing complaints about his work. To Schrag, the 2 disputed threads of alpha-beta research raise far-reaching questions about scientific integrity in the struggle to understand and cure Alzheimer's. Some adherents of the amyloid hypothesis are too uncritical of work that seems to support it, he says. Even if misconduct is rare, false ideas inserted into key nodes in our body of scientific knowledge can warp our understanding. In his modest office, steps away from a buzzing refrigerator, Schrag displays an antique microscope, an homage to predecessors who applied painstaking bench science to medicine's endless enigmas. A small sign on his desk reads, "Everything is figure-outable." So far, Alzheimer's has been an exception, but Schrag's background has left him comfortable with the field's contradictions. His father hails from a family of Mennonites, known for their philosophy of peacemaking, but joined the military. The family moved from Arizona to Germany to England before settling in Davenport, a tiny cow town in eastern Washington. After leaving the Air Force, Schrag's dad became a nurse and worked in a nursing home. As a young teen, Schrag volunteered to visit dementia patients there. I remember being mystified by a lot of the strange behaviors, he says. It was a formative experience to see people struggling with such unfair symptoms. Homeschooled by his mom, Schrag entered community college at 16, like many of the town's studious kids, including his teenage sweetheart and future wife, Sarah. They now live on a small ranch outside Nashville with their 2 young children and 3 aging horses that Sarah grew up with. While prepping for medical school at the University of North Dakota, Schrag spent long hours in a neuropharmacology lab absorbing the patient rhythms of science. He repeated experiments over and over, refining his skills. These included a protein identification method known as the Western blot. It uses electricity to drive protein-rich tissue samples through a gel that acts like a sieve to separate the molecules by size. Distinct proteins, tagged and illuminated by fluorescent antibodies, appear as stacked bands. In 2006, Schrag's first publication examined how feeding a high-cholesterol diet to rabbits seemed to increase alpha-beta plaques and iron deposits in one part of their brains. Not long afterward, when he was an MD-PhD student at Loma Linda University, another research group found support for a link between Alzheimer's and iron metabolism. Encouraged, Schrag poured his energy into trying to confirm the connection in people, and failed. The experience introduced him to a disquieting element of Alzheimer's research. With this enigmatic, complex disease, even careful experiments done in good faith can fail to replicate, leading to dead ends and unexpected setbacks. One of its biggest mysteries is also its most distinctive feature, The plaques and other protein deposits that German pathologist Alois Alzheimer first saw in 1906 in the brain of a deceased dementia patient. In 1984, alpha beta was identified as the main component of the plaques. And in 1991, researchers traced family-linked Alzheimer's to mutations in the gene for a precursor protein from which amyloid derives. To many scientists, it seemed clear that alpha-beta buildup sets off a cascade of damage and dysfunction in neurons, causing dementia. Stopping amyloid deposits became the most plausible therapeutic strategy. Where it's like, as far as I understand, the working model of Alzheimer's was, these amyloid plaques forming in the brain. That, the more and more they accumulate, the more damage done to the brain, and that, the more progressive Alzheimer's becomes, and then, you have increasing symptoms of Alzheimer's. Hundreds of clinical trials of amyloid-targeted therapies have yielded few glimmers of promise. However, only the underwhelming Aduhelm has gained FDA approval. Yet alpha-beta still dominates research and drug development. NIH spent about $1.6 billion on projects that mention amyloids in this fiscal year, about half its overall Alzheimer's funding. Scientists who advance other potential Alzheimer's causes, such as immune dysfunction or inflammation, complain they have been sidelined by the amyloid mafia. Forsyth says the amyloid hypothesis became the scientific equivalent of the Ptolemaic model of the solar system, in which the sun and planets rotate around the Earth. Where it's like, it seems like maybe, or potentially, based off of this research, we might have been working with the incorrect model of Alzheimer's for like the longest time, for like over a decade. And it seems like it may have come at the expense of, other researchers or, other hypotheses. Whereas they say, there are researchers trying to determine if there's, if immune dysfunction is playing a part in Alzheimer's, if inflammation is playing a part in Alzheimer's. And, that research, it seems like it wasn't funded as much because, this amyloid plaque model just became the dominant model. Like we just took it and ran with it, at the expense of other, maybe better models. And welcome back. By 2006, the centenary of Alois Alzheimer's epic discovery, a growing cadre of skeptics wondered aloud whether the field needed a reset. Then a breathtaking Nature paper entered the breach. It emerged from the lab of UMN physician and neuroscientist Karen Ash, who had already made a remarkable series of discoveries. As a medical resident at UCSF, she contributed to Nobel laureate Stanley Prusner's pioneering work on prions. And prions, while very cool, very damaging, but there's a little bit of overlap between prions and this amyloid plaque hypothesis, where it seems prions are basically, it's in the name, they are these what seem like infectious protein molecules, where it's like, if you have one sort of misfolded protein that somehow gets incorporated into your body, there's like this domino or cascading effect of like this misfolded protein causes the proteins next to it to misfold, and then next to those ones to misfold, and that seems to be very much at play in these prion diseases. Infectious proteins that cause rare neurological disorders. In the mid-1990s, she created a transgenic mouse that churns out human alpha-beta, which forms plaques in the animal's brain. The mouse also shows dementia-like symptoms. It became a favorite Alzheimer's model. Second, I just need a bit of a drink. And it seems like, you know, based off of this and what they're outlining, although I think this is a common thing, of like, we scientists, potentially myself included, we can be unscientific to a certain extent. Of like, it seems like we are very quick to latch on to either a model, or, a hypothesis, if it sounds or, proves useful. We're not necessarily the most concerned with, what is true or what is correct, but more like, oh, if this is useful, then let's just go with that. Where it's like, yeah, like, sure, this mouse model was able to display dementia-like symptoms, and then it looks like it became a favored Alzheimer's model. Where it's like, is this actually the best model for Alzheimer's? Probably not. But since this works, since probably other scientists adopted it, and it became convention, then it just probably came convention throughout all of Alzheimer's research. That's pretty common in science. Of like, if we're looking to do something, okay, if we're looking to like do an experiment, our first instinct is to like find out how some other scientists did the experiment, and then if it worked for them, well, then we go with that. Even though what has done in the past hasn't necessarily proved to be true, or the most optimal, but it's just been proven to work. And so if it's been proven to at the very least work or function, that's good enough for us, and then other researchers adopt it. What can the average person do to help hold scientists accountable or to help point out this is a fantastic question. And this is one that I'm interested in answering myself, because I'm not even sure I have a concrete answer for you, other than to this is sort of my attempt at holding scientists accountable. I think is to be willing to talk about the bad science, because and to be willing to talk publicly about the bad science, because I don't think, and understandably, I don't think the general public really knows, how much bad science gets done. I don't think retractionwatch.com is, I don't think the general public, regularly visits this website. And I think also to like, to be willing to talk publicly or openly about how, how science functions, or like how our scientific institutions function. It seems like it becomes a breeding ground, or like it almost incentivizes bad science, which I will attempt to cover, where it's like the incentives are all wrong. Sure, on paper, science, is about, search for truth, trying to understand the world, form hypotheses, generate conclusions, gather data, and, interpret data with rigor, and hopefully interpret data correctly, form conclusions, and, really trying to, get to the truth of a matter. But, that's how science is on paper, but that's not really how it functions in the real world, at an institutional level, like at the university level, where, not necessarily what's true that matters. It's like, what can you get published? Or like, what can lead to a profitable product down the road? Or like, what will be useful to humanity down the road? That is what, science is mostly concerned with, at an institutional level. And like, again, on paper, science, at least in my mind, is supposed to be a collaborative effort. Supposed to be, me and my fellow scientists working together to hopefully seek out the truth, understand the world, understand nature. But it doesn't actually operate like that in the real world, because at an institutional level, like, science is a competition, either first to publish, or like first to generate a result. Because like I can speak for myself in my own research. Like if there is like I am working on a specific research question, okay, and there may very well be another lab or another PhD student trying to answer the exact same question. And if that's the case, it becomes a race, okay? Even though it would be in everyone's interest, at least if we're concerned with truth, of like, oh, if I know something about my research question, and I have results, and I know this other researcher working on the same question, they have their results, it's in our interest, if we are actually concerned with truth, to pool our results together, saying, hey, show me the data that you have, and I'll show you the data that I have. But it doesn't work like that. In fact, my boss, my university would hate that, because they would say, like, you're undermining our institution, because we want to be the first to publish this new finding, and that if you just give it away, then like we're not getting the publication. Our name isn't getting the recognition, and so that's bad. So the problem is that it's a for-profit industry. Is that, yes, that's a fairly good way to summarize it. Not necessarily like for profit, but either for profit, status, ego, etc. Those types of things. Let's see. Yeah, right. By the early 2000s, toxic oligomers, subtypes of AB, alpha beta that dissolve in some bodily fluids, had gained currency as a likely chief culprit for Alzheimer's, potentially more pathogenic than the insoluble plaques. Amyloid oligomers had been linked to impaired communication between neurons in vitro and in animals, and autopsies have shown higher levels of the oligomers in people with Alzheimer's than in cognitively sound individuals, but no one had proved that any of the many known oligomers directly caused cognitive decline. In the brains of Ash's transgenic mice, the UMN team discovered a previously unknown oligomer species, dubbed alpha beta 56, pronounced amyloid beta star 56, after its relatively heavy molecular weight compared with other oligomers. The group isolated amyloid beta star 56 and injected it into young rats. The rat's capacity to recall simple, previously learned information, such as the location of a hidden platform in a maze, plummeted. The 2006 paper's first author, sometimes credited as the discoverer of amyloid beta 56, was Lesnay, a young scientist Ash had hired straight out of a PhD program at the University of Gien, Normandy in France. What can we do to mitigate this issue? Yeah, also, again, another fantastic question. I'm not sure I have a concrete answer to that either, other than to kind of like fully revamp how we fund science. And like, instead of publishing to journals, like, we should be publishing, to either some sort of public platform that people and go and visit themselves. Like, it makes no sense in terms of at the very least, generally speaking, in the United States and Canada, a large portion of the research that's conducted is publicly funded. Okay, it comes from taxpayers' pockets, and yet despite that, okay, like, the research, a lot of the research, if not the majority of the research that takes place at universities, like, it's not available to the public. Like, it's hidden behind a paywall. Where it's like, I don't want to get too far away. But like, it should just be kind of like a public reporting system of like, hey, and also like to at least to be able to show where the tax dollars are going, and that, oh, this is research publicly funded, and like if the public, or if anybody is interested in like tracking the results of, you know, a researcher who's publicly funded, like they should be able to, you know, have somewhere to go online, be like, oh, this is the results that they've published so far, as opposed to like, you know, it's in a journal, that, you know, if you want to know the results or the conclusions, you know, you got to pay like whatever, 50, $100 just to like access the article, even though, like, you know, the public has funded it themselves. That doesn't make any sense, at least not to me. I think that's like the one place I would start. There's that alone, it's not going to solve it, but I think that would be a pretty big step in the right direction. Ash touted amyloid beta star 56 on our website as the first substance ever identified in brain tissue in Alzheimer's research that has been shown to cause memory impairment. An accompanying editorial in Nature called amyloid beta star 56, a star suspect in Alzheimer's. Alts Forum, a widely read online hub for the field, titled its coverage, Amyloid Beta Star is Born, less than 2 weeks after the paper was published. Ash won the prestigious Potamkin Prize for Neuroscience, partly for work leading to amyloid beta star 56. After the Kansas vote is in, 64-36 in favor of keeping abortion. Ooh, that's reassuring. And also, thank you so much for the update, Bob Marley. Super appreciate it. Can always count on you for good reporting, good data, sharing cool studies. It's almost like how the insurance industry hijacked the pharmaceutical industry. The journals and publishers put a huge bottleneck. Yeah, like scientific journals, like they are just like ridiculous. Don't get me started on those. Like, you know, the amount of profit that these scientific journals make is ridiculous, and there's no reason why it should be like that. Their profit margins are like, you know, the 30, 40, 50% range, which I think is like pretty much unheard of in any other industry. The Nature paper has been cited in about 2,300 scholarly articles, more than all but 4 other Alzheimer's basic research reports published since 2006, according to the Web of Science database. Since then, annual NIH support for studies labeled amyloid, oligomer, and Alzheimer's has risen from near zero to $287 million in 2021. Lesney and Ash helped spark that explosion, experts say. The paper provided an important boost to the amyloid and toxic oligomer hypotheses when they faced rising doubts. Proponents loved it because it seemed to be an independent validation of what they have been proposing for a long time. That was a really big finding that kind of turned the field on its head, partly because of Ash's impeccable imprimatur, Wilcox says. It drove a lot of other investigators to go looking for these heavier oligomer species. As Ash's star burned more brightly, Lesney's rose. He joined UMN with his own NIH-funded lab in 2009. Amyloid beta star 56 remained a primary research focus. Megan Larson, who worked as a junior scientist for Lesney and is now product manager at Biotechny, a biosciences supply company, calls him passionate, hardworking, and charismatic. She and others in the lab often ran experiments and produced Western blots, Larson says, but in their papers together, Lesney prepared all the images for publication. He became a leader of UMN's neuroscience graduate program in 2020, and in May 2022, not that long ago, 4 months after Schrag delivered his concerns to NIH, Lesney received a coveted R01 grant from the agency with up to 5 years of support. And this is like, from what I understand, a very huge grant. Like in, I think, the millions of dollars. Probably not Or like, yeah, close to 1,764,000. So, not small change, or not a small amount when it comes to scientific funding. The NIH program officer for the grant, Austin Yang, a co-author of the 2006 Nature paper, declined to comment. In December 2021, Schrag visited PubPeer, a website where scientists flag possible errors in published papers. So this might be another website that might be flagging fraudulent research. We have Retraction Watch, that's the one that I'm most familiar with. But it seems like you also have PubPeer. Whoops. Where it's like, it seems like this is devoted to flagging maybe problematic research or possible errors. Many of the site's posts come from technical gumshoes who deconstruct Western blots for telltale marks, indicating that bands representing proteins could have been removed or inserted where they don't belong. Such manipulations can falsely suggest a protein is present, or alter the levels at which a detected protein is apparently found. Bragg, still focused on cassava-linked scientists, was looking for examples that could refine his own sleuthing. And well, yeah, this is also a Western blot. And you can see that they are showing the example of what I tried to go through, of like, when we report about certain types of data, we don't necessarily show the full result. It's sort of standard practice that, we zoom in onto certain area of interest, and that, we just go on faith when we submit this to journals that, all the stuff that you're not seeing, well, it's all good. We're not doing anything fraudulent, or we're not being deceptive. Just trust us that what we're zooming in on is correct, and that there's nothing to see in the rest of the image. In a PubPeer search for Alzheimer's, postings about articles in the Journal of Neuroscience caught Schrag's eye. They questioned the authenticity of blots used to differentiate amyloid beta and similar proteins in mouse brain tissue. Several bands seemed to be duplicated using software tools. Schrag confirmed the PubPeer comments and found similar problems with other blots in the same articles. He also found some blot backgrounds that seemed to have been improperly duplicated. How an image sleuth uncovered possible tampering. Vanderbilt University neuroscientist Matthew Schrag found apparently falsified images in papers by University of Minnesota, Twin Cities, neuroscientist Sylvain Lesne, including, So basically, image manipulation. I'm not going to dissect this portion here, but they're basically showing there appears to be doctored images in the original publication. 3 of the papers listed Lesne, whom Schrag had never heard of, as first or senior author. Schrag quickly found that another Lesne paper had also drawn scrutiny on PubPeer, and he broadened his search to Lesne papers that had not been flagged there. The investigation developed organically, he says, as other apparent problems emerged. So much in our field is not reproducible, so it's a huge advantage to understand when data stream might not be reliable, Schrag says. Some of that's going to happen reproducing data on the bench, but if it can happen in simpler, faster ways, such as image analysis, it should. Eventually, Schrag ran across the seminal Nature paper, the basis for many others. It too seemed to contain multiple doctored images. Science asked 2 independent image analysts, Vic and Jana Christopher, to review Schrag's findings about that paper and others by Lesne. They say some supposed manipulation might be digital artifacts that can occur inadvertently during image processing, a possibility Schrag concedes. But Bick found his conclusions compelling and sound. Christopher concurred about the many duplicated images and some markings suggesting cut-and-pasted Western blots flagged by Schrag. He also identified additional dubious plots and backgrounds he had missed. Do journals genuinely just ignore that, or will they require some kind of proof if you do something pretty significant or unbelievable? It's, as far as I know, it's pretty rare. Like, unless there's something glaringly obvious when you submit something to peer review, they are not really going to question it. As in, I could even use my own research as maybe as an example. Where it's like, you know, this is data that I had a hand in generating, played a part in. This is just a, you know, textbook figure. But you know, there's like, you know, some fancy, well, arguably, you know, fancy graphs. And this was generated by a particular instrument. And you know, we submitted these graphs. But like, these are graphs generated by, like, you know, an instrument measuring a sample, okay, without, you know, getting into the details of like how. But you could easily like make these in Excel or whatever, some other graphing software. Like, right? This is not, it would not be super complicated to like generate a similarly shaped graphs in Excel, right? It's just a bunch of like curves. As in, like, especially like these ones. You know, I'm sure if I tasked just about anyone that has a decent knowledge of Excel to like recreate something similar to this, they'd be able to do it pretty quickly. But it's just sort of, you know, it's just things are taken at face value. Of like, oh, if this is the data, these are the graphs, we just assume that this is data generated from like measuring a sample. Oh wow, thank you so much for the sub, Tyler, and also thank you so much for passing by. Super appreciated. Always happy to have you here, and you know, can't thank you enough for your support. But yeah, you know, to pretty much answer your question as best I can, Bob Marley, like journals, they don't do a lot of vetting when data are submitted to them, just because to do that, like, it's a huge undertaking. Like, if peer reviewers wanted to actually like wanted us to prove that, you know, oh, these graphs, it's coming from, you know, a sample that's being measured in an instrument, like, I have no problem proving that, but it would take so much time and effort on their part to like go through all that stuff. And peer reviewers, like, they work on a voluntary basis, right? They're not paid. And so it's like, you know, why are they going to waste their voluntary time, you know, going above and beyond, ensuring that only, you know, high quality, true research gets conducted. In the 16 years following the landmark paper, Lesnay and Ash, separately or jointly, published many articles on their stellar oligomer. Yet only a handful of other groups have reported detecting amyloid beta star 56. Citing the ongoing UMN review of Lesnay's work, Ash declined via email to be interviewed or to answer written questions posed by Science, which she called sobering, which is a little, I don't know, a little suspicious. Where if, like, if a particular scientist's work is being questioned, and, you know, people are reaching out to comment, and they're like, I don't want to comment, I think that's pretty suspect. I still have faith in amyloid beta star 56, noting her ongoing work studying the structure of amyloid beta oligomers. We have promising initial results. I remain excited about this work and believe it has the potential to explain why amyloid beta therapies may yet work despite recent failures targeting amyloid plaques. But even before Schrag's investigation, this body evidence that amyloid beta star 56 plays a role in Alzheimer's had raised eyebrows. Wilcock has long doubted studies that claim to use purified amyloid beta star 56. So I'm curious, what makes you say that? Like, I mean, at the end of the day, everyone has the right to, you know, refuse to comment. Well, what I would only say, like, my response would be, if, let's say, if my own research was like being called into question, and like, they wanted me to comment on it, like I'm more than happy to defend my own research if I'm being called out. Obviously, like, no one's gonna call out my research because, like, it's obscure and not a lot of people in the world care about it. But at least me as a researcher, like, if my research was being called into question, like, I would personally want to say something and, like, stand up for my own research. But I could be wrong. I might be the outlier. Such oligomers are notoriously unstable, converting to other oligomer types spontaneously. Multiple types can be present in a sample even after purification efforts, making it hard to say any cognitive effects are due to amyloid beta star 56 alone. She notes, assuming it exists. In fact, Wilcock and others say several labs have tried and failed to find alpha beta star 56, although few have published those findings. Journals are often uninterested in negative results, and researchers can be reluctant to contradict a famous investigator. And here's another, like, huge issue in science, and another reason as to why, okay, I would just generate sort of like open science of, like, you know, scientists, instead of publishing to, you know, for-profit journals, like, we're just like uploading to whatever, some sort of hopefully organized database, you know, just like showing the results that's accessible to the public. Because with this type of open science, then you can publish, or you can at least, like, show negative results. Because as they highlight here, and they highlight correctly, okay, journals are often uninterested in negative results. As in, like, if you attempt to do an experiment, okay, and it doesn't work out, well, that's not really publishable. You know, Science or Nature journal is not gonna, you know, be all enthusiastic of, like, oh, you tried this and it failed? Well, let's get it on in the books as quickly as possible. As opposed to, like, with open science, where it's like, you know, publicly accessible database. Like, you can upload that. Not only can the general public see it and access it, but other scientists can see it and access it. And that is useful information. Like, in science, basically, like, the most useful information is knowing what doesn't work. Because it's generally speaking, the super rare situation that something actually works out in science. Like, the vast majority of science, like 80-90%, fails. And so, technically speaking, we are not publishing like 80 to like 90% of science. And I think that's a huge problem with huge consequences. Like, literally, for all I know, someone else has already attempted what I'm attempting to answer, but they failed. But since that is not published, I don't know about that. Like, literally, someone, I could be literally replicating what someone else, some other scientist has done, not know about it, because negative results are not published. Or it's exceedingly rare when negative results are published. Because if you respond to the allegations in the modern political climate, people will take any kind of, sure, yeah, that's fair. I can totally understand that perspective. Let's see. An exception was Harvard University's Dennis Selke, a leading advocate of the amyloid and toxic oligomer hypotheses, who has cited the Nature paper at least 13 times. In two 2008 papers, Selke said he could not find alpha amyloid beta star 56 in human fluids or tissues. Selke examined Schrag's dossier on Lesnay's papers at Science's request and says he finds it credible and well supported. He did not see manipulation in every suspect image, but says there are certainly at least 12 or 15 images where I would agree that there is no other explanation than manipulation. 1. An image in the Nature paper displaying purified amyloid beta star 56 shows very worrisome signs of tampering. Selke says. The same image reappeared in a different paper, co-authored by Lesnay and Ash 5 years later. Many other images in Lesnay's papers might be improper, more than enough to challenge the body of work, Selke adds. A few of Lesnay's question papers describe a technique he developed to measure amyloid beta oligomers separately in brain cells, bases outside the cells, and cell membranes. Selke recalls Ash talking about her brilliant postdoctoral fellow who devised it. He was skeptical of Lesnay's claim that oligomers could be analyzed separately inside and outside cells in a mixture of soluble material from frozen or processed brain tissue. All of us who heard about that knew in a moment that it made no biochemical sense. If it did, we'd all be using a method like that. The Nature paper depended on that method. Selke himself co-authored a 2006 paper with Lesnay in the Annals of Neurology. They sought to neutralize the effects of toxic oligomers, although not amyloid beta star 56. The paper includes an image that Schrag, Bick, and Christopher agree was reprinted as if original in 2 subsequent Lesnay articles. Selke calls that highly egregious. Given those findings, the scarcity of independent confirmation of the amyloid beta star 56 claims seems telling, Selke says. In science, once you publish your data, if it's not readily replicated, then there's real concern that it's not correct or true. There's precious little clear-cut evidence that amyloid beta star 56 exists, or if it exists, correlates in a reproducible fashion with features of Alzheimer's even in animal models. In all, Schrag or Bick identified more than 20 suspect Lesnay papers, 10 concerned amyloid beta star 56. Schrag contacted several of the journals starting early this year, and Lesnay and his collaborators recently published 2 corrections, one for a 2012 paper in the Journal of Neuroscience, replaced several images Schrag had flagged as problematic, writing that the earlier versions had been processed inappropriately. But Schrag says even the corrected images show numerous signs of improper changes in bands, and in one case, complete replacement of a blot. To clarify, is the point of this stream to finish reading the article, or is it something else? If it's the former, well, first of all, nothing to apologize for. And to answer your question, like, I have multiple goals for this stream. Like, one is covering this topic, okay? And like, I don't have a timeline. Like, I want to finish this article, although it does seem to be pretty lengthy. But I am mostly done. And then from there, cover some other topics. There's some YouTube videos that I wanted to watch as well on a bunch of various topics. I think mostly political content. So hopefully that answers your question. Where was I? I was past here. I think I was past there. Oh, no, I was right here. So, yeah, after Science contacted Ash, she separately posted to PubPeer a defense of some images Schrag had challenged in the Nature paper. She supplied portions of a few original unpublished versions that do not show the apparent digital cut marks Schrag had detected in the published images. That suggests the markings were harmless digital artifacts. Yet, the original images reveal something that Schrag and Selke find even more incriminating. Unequivocal evidence that despite the lack of obvious cut marks, multiple bands were copied and pasted from adjacent areas. Schrag could find no innocent explanation for a 2-decade litany of oddities. In experiment after experiment using Western blots, microscopy, and other techniques, serious anomalies emerged. But he notes that he has not examined the original uncropped high-resolution images. Authors sometimes share those with researchers conducting similar work, although they usually ignore such requests. According to recent studies of data sharing practices, sharing agreements do not include access for independent misconduct detectives. Lesney and Ash did not respond to a science request for those images. Questions about Lesney's work are not new. Cell biologist Dennis Vivian, a senior scientist at Kane, co-authored 5 Lesney papers flagged by Schrag or Bick. Vivian defends the validity of those articles, but says he had reason to be wary of Lesney. Toward the end of Lesney's time in France, Vivian says they worked together on a paper for Nature Neuroscience involving amyloid beta. During the final revisions, he saw immunostaining images in which antibodies detect proteins in tissue samples that Lesney had provided. They looked dubious to Vivian, and he asked other students to replicate the findings. Their efforts failed. Vivian says he confronted Lesney, who denied wrongdoing. Although Vivian lacked irrefutable proof of misconduct, he withdrew the paper before publication to preserve my scientific integrity and broke off all contact with Lesney. He says, "We are never safe from a student who would like to deceive us, and we must remain vigilant." Schrag spot-checked papers by Vivian or Ash without Lesney. He found no anomalies, suggesting Vivian and Ash were innocent of misconduct. Yet senior scientists must balance the trust essential to fostering a protege's independence with prudent verification, Wilcox says. If you sign off on images time after time, claim credit, speak publicly, and win awards for the work, as Ash has done, you have to be sure it's right, she adds. Ash obviously failed in that very serious duty to ask tough questions and ensure the data's accuracy, Forsyth says. It was a major ethical lapse. In his whistleblower report to NIH about Lesney's research, Schrag made its scope and stakes clear. This dossier is a fraction of the anomalies easily visible on review of the publicly accessible data, he wrote. The suspect work not only represents a substantial investment in NIH research support, but has been cited thousands of times and thus has the potential to mislead an entire field of research. The agency's reply, which Schrag shared with Science, noted that complaints deemed credible will go to the Department of Health and Human Services Office of Research Integrity for review. That agency could then instruct grantee universities to investigate prior to a final ORI review, a process that can take years and remains confidential absent an official misconduct finding. To Science, NIH said it takes research misconduct seriously, but otherwise declined to comment. In the fanfare around the Lesney-Ash work, some Alzheimer's experts see a failure of skepticism, including by journals that publish the work. After Schrag contacted Nature, Science Signaling, and 5 other journals about 13 papers co-authored by Lesney, a few are under investigation, according to emails he received from editors. There are very strong legitimate questions, John Foley, editor of Science Signaling, later told Science. He says the journal has contacted authors and university officers of 2 papers from 2016 and 2017 for a response. It also recently issued expressions of concern about the articles. A spokesperson for Nature, which publishes image integrity standards, says the journal takes concerns raised about its papers seriously, but otherwise had no comment. Days after an inquiry from Science, Nature published a note saying it was investigating Lesney's 2006 paper and advising caution about its results. The Journal of Neuroscience stands out with 5 suspect Lesney papers. A journal spokesperson said it follows guidelines from the Committee on Publication Ethics to assess concerns, but otherwise had no comment. Journals and granting institutions don't know how to deal with image manipulation, Forsyth says. They're not subjecting images to sophisticated analysis, even though those tools are very widely available. It's not some magic skill. It's their job to do the gatekeeping. You can't cheat to cure a disease. Biology doesn't care. So, like, yeah, pretty in-depth, and a good, I think, assessment of the events. And I guess now the ongoing investigation. It seems like multiple articles from this scientist's work is being called into question, and it looks like there's active investigations taking place at these journals where this scientist published. So I will be tracking that. Before we get into watching some, what I hope be interesting YouTube content, just to touch upon sort of bad incentives. This short article sort of touches upon that. It's about a bad system, fraud and fabrication in scientific research. How competition and limited funding can lead to dishonesty. Where, like, sort of as I said previously, that, science, generally speaking, is not collaborative. At least when it comes to, boots on the ground science. It's a competition. Competition to publish, competition to generate results, competition for scholarships, competition for money. And so, the incentive to cheat or, to potentially get ahead is built in to the system. On July 21st, an article in Science magazine questioned the integrity of a key Alzheimer's study, sending shockwaves through the world of science and academia. The allegations lodged against a University of Minnesota research team center around a claim that one of the researchers, Sylvain Lesnay, falsified images used in the study. The study has been foundational in understanding Alzheimer's and treatment development for the disease. Lesnay did not respond to a request for comment. Colleague Karen Ash said in an email she is devastated that Lesnay may have misled her and the scientific community. She said she is working with the journal to rectify the situation, but that the research findings stand even without those images. The University of Minnesota told us it will follow its processes to review the questions any claims have raised. To dig deeper into scientific fraud and misconduct and why it occurs, NPR News host Tom Gran spoke with Ray DeVries, professor emeritus at the Center for Bioethics and Social Sciences in Medicine at the University of Michigan. This is basically sort of a transcript or interview. When you heard about this situation at the University of Minnesota, what did you think? I thought this is nothing new. This is an example of the problem we have with how we organize and pay for science in the United States, and in Canada also. Several years ago, we did research on what are the things associated with people who are, let's say, dishonest in their scientific research. Especially when you look at this case, it's like, oh, these researchers must be bad people. But we had a suspicion that it was something more about a bad system. In the United States, we believe, essentially, the best way to get the best science is to have scientists compete with each other. And what we found is that when scientists compete, it actually leads them to cut corners because it was in the interest of the scientists to continue getting funding for their research. Which is pretty much my sentiment exactly and what I observe. Where, we are all under this notion, or not all, but it seems like, the popular notion that, competition is what drives things forward. Competition drives innovation. Competition drives economic progress. When even though there's like no evidence that that's the case, and we have increasing evidence that this notion of competition sort of undermines many things and undermines doing good science. Because like if you're competing with your neighbor, especially if that you're competing with your neighbor for like funding, for access to research fundings, well then kind of all the more reason to cheat or cut corners. Tell us more about the spectrum of what you see when it comes to scientific fraud and what you refer to as cutting corners for competitive advantage. If you look at the whole science industry, you want to be the first one to report findings on some significant new breakthrough. So you're looking at your data and you think, well, if I just eliminate an outlier, my data will be cleaner. It'll make a stronger case, more likely to be published. And if I get published, it's more likely I'll get my next grant. Then you slip into things like, as we've seen in this case, apparently manipulating images to make the hypothesis you believe in look more true than it really is. If the allegations in this case at the University of Minnesota are true, what impact does that have on research, specifically on Alzheimer's and more generally? Let's say only 1% of all researchers engage in these kinds of behaviors. When the data that they're creating gets into the scientific record, it has this huge effect. So scientists start using that as their starting point, and it's changing the entire direction of scientific research. I think that's the case we've seen here. 16 years of research has been based on a paper that may have been using falsified data. If it's shown that these are in fact falsified data, the paper will be retracted, but that paper lives on, especially now in the era of the internet. What needs to change in order to prevent something like this from happening in the future? The key thing here is realizing that we have to change the reward structure of science. Because we reward scientists for cutting-edge discoveries, we don't reward them for doing replication studies. And this is another important fact, that if you look at funding from the National Institutes of Health over the last 20 years, it's essentially been flat. This means that we have a number of trained scientists out there competing for the same pot of money that's staying the same size. This creates the problem of, in this competitive atmosphere, why would I share my data if I'm at Minnesota with a lab at Michigan, because they might get the next grant? Some people suggest the creation of research consortia where all these laboratories come together, and when one lab makes an advancement, all the lab gets the information, and they carry that on. When people hear something like this, what do they need to keep in mind? Science has ways, as shown in this case, of being self-correcting. I think we need to trust that the majority of scientists are trustworthy. They are checking on each other. If anything, we need more efforts to promote cooperation, rather than the kind of competition that leads to certain scientists saying, I'll do anything to get the next grant. So yeah, this pretty much sums up all of my sentiments of like this atmosphere of competition, like, is pretty much overall detrimental to all of science. Oh, and rhinos are unicorns. Welcome. Thank you so much for passing by. Always happy to have you here. Super grateful. So yeah, now hopefully do some YouTube.
Do you...
So, I think this is maybe a good YouTube video to, address, the 4 things you need to be an expert. What is expertise? I think that's a broad philosophical question. Well, I'm glad you've been sharing it, because, even though I'm a scientist, like, I want bad science, or fraudulent science to be exposed. Because, like, I want, not that science will ever be perfect, but I'm obviously not in favor of bad or fraudulent science. And that we need to weed out the bad actors.
Bring this trick out at parties?
Oh no, it's a terrible party trick. Here we go. 3.141592653589793.
This is Grant Gussman. He watched an old video of mine about how we think, that there are 2 systems of thought. System 2 is the conscious, slow, effortful system, and System 1 is subconscious, fast and automatic. To explore how these systems work in his own head, Grant decided to memorize 100 digits of pi.
38446.
Then, he just kept going. He has now memorized 23,000 digits of pi in preparation to challenge the North American record.
8954930388196. That's 200.
That's amazing. I have wanted to make a video about experts for a long time. This is Magnus Carlsen, the 5-time world chess champion. He's being shown chessboards and asked to identify the game in which they occurred.
This looks an awful lot like Tal Botvinnik. Whoops. Okay, this is the 24th game from Seville, obviously.
Now I'm going to play through an opening and stop me when you recognize the game, and if you can tell me who was playing black in this one.
Okay, sure.
Okay. So I'm sure you've seen this opening before.
Okay, it's gonna be Anand.
Against
Zapata.
How can he do this? It seems like superhuman ability. Well, decades ago, scientists wanted to know what makes experts like chess masters special. Do they have incredibly high IQs? Much better spatial reasoning than average? Bigger short-term memory spans? Well, it turns out that as a group, chess masters are not exceptional on any of these measures. But one experiment showed how their performance was vastly superior to amateurs. In 1973, William Chase and Herbert Simon recruited 3 chess players: a master, an A player, who's an advanced amateur, and a beginner. A chessboard was set up with around 25 pieces positioned as they might be during a game, and each player was allowed to look at the board for 5 seconds. Then they were asked to replicate the setup from memory on a second board in front of them. The players could take as many 5-second peeks as they needed to get their board to match. From just the first look, the master could recall the positions of 16 pieces. The A player could recall 8, and the beginner only 4. The master only needed half the number of peeks as the A player to get their board perfect. But then the researchers arranged the board with pieces in random positions that would never arise in a real game. And now, the chess master performed no better than the beginner. After the first look, all players, regardless of rank, could remember the location of only 3 pieces. The data are clear. Chess experts don't have better memory in general, but they have better memory specifically for chess positions that could occur in a real game. The implication is what makes chess masters special is that they have seen lots and lots of chess games. And over that time, their brains have learned patterns. So rather than seeing individual pieces at individual positions, they see a smaller number of recognizable configurations. This is called chunking. What we have stored in long-term memory allows us to recognize complex stimuli as just one thing. For example, you recognize this as pi rather than a string of 6 unrelated numbers, or meaningless squiggles for that matter.
There's a wonderful sequence I like a lot, which is 30173, which to me means Stephen Curry, number 30, 173 games, which is the record back in 2016, so 30173.
At its core, expertise is about recognition. Magnus Carlsen recognizes chess positions the same way we recognize faces. And recognition leads directly to intuition. If you see an angry face, you have a pretty good idea of what's going to come next. Chess masters recognize board positions and instinctively know the best move.
Most of the time, I know what to do. I don't have to figure it out.
To develop the long-term memory of an expert takes a long time. 10,000 hours is the rule of thumb popularized by Malcolm Gladwell, but 10,000 hours of practice by itself is not sufficient. There are 4 additional criteria that must be met. And in areas where these criteria aren't met, it's impossible to become an expert. So the first one is many repeated attempts with feedback. Tennis players hit hundreds of forehands in practice. Chess players play thousands of games before they're grandmasters, and physicists solve thousands of physics problems. Each one gets feedback. The tennis player sees whether each shot clears the net and is in or out. The chess player either wins or loses the game, and the physicist gets the problem right or wrong. But some professionals don't get repeated experience with the same sorts of problems. Political scientist Philip Tetlock picked 284 people who make their living commenting or offering advice on political and economic trends. This included journalists, foreign policy specialists, economists, and intelligence analysts. Over 2 decades, he peppered them with questions like, would George Bush be re-elected? Would apartheid in South Africa end peacefully? Would Quebec secede from Canada? And would the dot-com bubble burst? In each case, the pundits rated the probability of several possible outcomes, and by the end of the study, Tetlock had quantified 82,361 predictions. So, how did they do? Pretty terribly. These experts, most of whom had postgraduate degrees, performed...
Yeah, I don't know how I feel about that. As I said before, I don't know. I have mixed feelings about economists. Not you, Bob Marley, but just other economists.
Worse than if they had just assigned equal probabilities to all the outcomes. In other words, people who spend their time and earn their living studying a particular topic produce poorer predictions than random chance. Even in the areas they knew best, experts were not significantly better than non-specialists. The problem is, most of the events they have to predict are one-offs. They haven't had the experience of going through these events or very similar ones many times before. Even
Oh, I agree. Obviously, I would never say all economists are bad or bad people. There are totally some good economists. But it seems like, I don't know. The field of economics like seems to attract a certain sort of person. Sometimes, not that everyone who studies economics are the same, but there's a certain sort of person that seems to gravitate towards economics. And yeah, to answer your question, anosmia. I was alive when that referendum happened, when Quebec like literally voted on whether or not they want to separate from Canada. And it was like, it was referendum night, and like it was like the slimmest of margins. Like it was literally like 50.5% staying with Canada, and like 49.5 seceding from Canada. And that was like a relief. I'm generally not in favor of Quebec separating from Canada. Yes, they are a very particular individual.
Presidential elections only happen infrequently, and each one in a slightly different environment. So we should be wary of experts who don't have repeated experience with feedback. The next requirement is a valid environment, one that contains regularities that make it at least somewhat predictable. A gambler betting at the roulette wheel, for example, may have thousands of repeated experiences with the same event, and for each one they get clear feedback in the form of whether they win or lose. But you would rightfully not consider them an expert because the...
Yeah, actually, to corroborate my story, yeah, the no option, as in, no, not separating from Canada, carried by 50.5%. So again, 50.58%. So like, again, the narrowest of margins. I remember that night of, some people like really being on the edge of their seat. I'm like, oh, if Quebec separates from Canada, like, we're out of here. Or like, yeah, you see the breakdown here. Where, yeah, 50.58, 49.42%. Yes, separate. Like, I don't think it gets any more close than that. A knife's edge. And I don't think Quebec could sustain itself. I mean, already Quebec doesn't sustain itself, like, and doesn't seem like any province does, because we receive, transfer payments from the federal government, and Quebec receives a lot of money from the federal government. And yeah, I don't necessarily disagree with you. There's actually, maybe we should watch some of these videos somewhere down the line, where there's a channel called Unlearning Economics, and he has a whole video devoted to the toxic culture of the economics profession, and they're an economist, from what I can tell. Yeah, I'm an academic economist from the UK. And he pretty much has a whole video pretty much like bashing at least the culture of the economics profession. But I will totally agree with you that there are some economic studies that I certainly appreciate and respect 100%, and there's some good research that has been conducted by economists, for sure. I won't deny that. I have actually. That was like one of the first videos I watched, like, or the second. The first one was this Jordan Peterson one. Not surprisingly, they're, not a fan of Jordan Peterson. And he pretty much goes through the same thing that I go through, of like, Jordan Peterson doesn't know what he's talking about, doesn't seem to know how to analyze science. And then I did watch this one, but I have no problem watching it again. Maybe we'll watch it, or rather, we will watch this on stream in the future. Maybe my next stream, on Sunday.
The environment is low validity. A roulette wheel is essentially random, so there are no regularities to be learned. In 2006, legendary investor Warren Buffett offered to bet $1 million that he could pick an investment that would outperform Wall Street's best hedge funds over a 10-year period. Hedge funds are pools of money that are actively managed by some of the brightest and most experienced traders on Wall Street. They use advanced techniques like short selling, leverage, and derivatives in
Well, obviously, you know, I can't compare it to anything because I only know my disciplines, which is biology, biochemistry, basically the life sciences. And, you know, those are, these fields are very much women dominated. So I think that changes the dynamics. I haven't really experienced any toxicity. I have observed toxicity, mostly coming from other men in these fields. And it's mostly in the form of, you know, basically sexual misconduct. But to kind of, you know, make my case that it, you know, there does seem to be a certain type of person that goes into economics. Like I've showed the studies before of, like, you know, it seems like economists, you know, they donate to charity less. It seems like on the whole, it seems like, you know, they're more willing to cheat at a game or like do things for their own selfish interest, more than other people. This is obviously not all economists, but you know, the actual data doesn't like paint economists like as all that, you know, altruistic, I would say.
In an attempt to provide outsized returns, and consequently they charge significant fees. One person took Buffett up on the bet, Ted Seides of Protégé Partners. For his investment, he selected 5 hedge funds. Well, actually, 5 funds of hedge funds, so in total a collection of over 200 individual funds. Warren Buffett took a very different approach. He picked the most basic, boring investment imaginable, a passive index fund that just tracks the weighted value of the 500 biggest public companies in America, the S&P 500. They started the bet on January 1st, 2008, and immediately things did not look good for Buffett. It was the start of the global financial crisis and the market tanked. But the hedge funds could change their holdings and even profit from market falls, so they lost some value but not as much as the market average. The hedge funds stayed ahead for the next 3 years, but by 2011 the S&P 500 had pulled even. And from then on, it wasn't even close. The market average surged, leaving the hedge funds in the dust. After 10 years, Buffett's index fund gained...
Actually, I'm curious. Sorry. What is the gender breakdown, obviously in the United States, of I believe third column, female. Yeah, it is. Just to satisfy my own curiosity. This. Yeah. So look at this. Economics general. Well, first of all, it seems to be like a super popular field. Super popular field for men. Where you have 20,000 males, per roughly 9,000 females. Like, I don't know. That's a little bit weird to me. Where it's like just about every other field, like this is like one of the few fields that is still male dominated. As opposed to, you know, you look at you look at biology or biological sciences, it's like 2 to 1 women. 47,000 to 26,000. And that holds true for pretty much all of the life sciences. Biochemistry, about 50-50. Molecular biology, generally women dominant. Structural biology. Well, there's no structural biologists apparently. And then microbiology and immunology, also women dominant. Yeah. Accounting, good. Yeah, so about 50-50. It look almost exactly split. 50-50, right? I think that's super interesting. Really super interesting. Where it's like, why are, men like flocking to economics? Like in droves. Like even when men are going to university, like, which is not a lot of them, they're just like, well, let's just go into economics. You know, whether there's a correlation between this being dominated by men and the perceived toxicity, I can't say with any sort of like high confidence. But let's just say, is it surprising to me that a discipline that is largely dominated, heavily dominated by men, is toxic? Then no, that doesn't surprise me. I think there's something deeper here than just, men just like economics. But that is speculation on my part. Admit that.
$125.8% to the hedge fund's 36%. Now, the market performance was not unusual over this time.
And my sister is an accountant, a full chartered accountant, and she actually teaches accounting.
At 8.5% annual growth, it nearly matches the stock market's long-run average. So why did so many investment professionals with years of industry experience, research at their fingertips, and big financial incentives to perform fail to beat the market? Well, because stocks are a low validity environment. Over the short term, stock price movements are almost entirely random. So the feedback, although clear and immediate, doesn't actually reflect anything about the quality of the decision making. It's closer to a roulette wheel than to chess. Over a 10-year period, around 80% of all actively managed investment funds failed to beat the market average. And if you look at longer time periods, underperformance rises to 90%. And before you say, well, that means 10% of managers have actual skill, consider that just through random chance, some people would beat the market anyway. Portfolios picked by cats or...
Maybe, but I think there's also, I think there's an interplay between those two things. As in, I think that competitiveness sort of, I think, fosters or cultivates a certain element of toxicity.
Or throwing darts have been shown to do just that. And in addition to luck, there are nefarious practices, from insider trading to pump and dump schemes. Now, I don't mean to say there are no expert investors. I mean, Warren Buffett himself is a clear example. But the vast majority of stock pickers and active investment managers do not demonstrate expert performance because of the low validity of their environment. Brief side note. If we know that stock picking will usually yield worse results over the long term, and that what active managers charge in fees is rarely compensated for in improved performance, then why is so much money invested in individual stocks, mutual funds, and hedge funds? Well, let me answer that with a story. There was an experiment carried out with rats and humans, where there's a red button and a green button that can each light up. 80% of the time, the green button lights up, and 20% of the time, the red button lights up, but randomly, so you can never be sure which button will light. And the task for the subject, either rat or human, is to guess beforehand which button will light up by pressing it. For the rat, if they guess right, they get a bit of food, and if they guess wrong, a mild electric shock. The rat quickly learns to press only the green button and accept the 80% win percentage. Humans, on the other hand, usually press the green button, but once in a while they try to predict when the red light will go on, and as a result, they guess right only 68% of the time. We have a hard time accepting average results, and we see patterns everywhere, including in randomness, so we try to beat the average by predicting the pattern.
Well, It's how come when one field is male dominated, we take it as male toxicity, but when a field is female dominated, we don't associate with female toxicity. So you're asking totally valid question. Okay. For sure. That is a legit question. And I will attempt to answer it as best I can. Because I think there's good reasons to make the argument that male toxicity or toxic masculinity is a thing. And that, sure, can women also be toxic? Yes, absolutely. But I think it's to a much less degree, much less significant, and that generally speaking, there is a far less chance of, if a field is being dominated by women, there's less chance of, having an environment that's toxic being cultivated. Not impossible. And I think there are good reasons for that. Good sociological reasons for that. I don't think this is some sort of trait inherent to women or, femme-presenting people or more feminine people. But generally speaking, on the whole, okay, women or femme-presenting people, they tend to be more cooperative, as in they tend to be more altruistic. They tend to be less selfish. Things like charity work. Charity work is generally mostly done by women because they tend to be more pro-social, as opposed to this culture of competitiveness seems to really be perpetuated in male-dominated or masculine spheres. And again, I think there's good sociological reasons for that. I'm not saying this is something biologically related to men. But the more pro-social or more cooperative nature of women, on average, is likely driven by multiple factors. One of them being empathy. Okay, we know that empathy can drive altruistic behavior or more pro-social behaviors. And if you measure the empathy levels of women, they are generally, on average, on the whole, way higher than men or masculine presenting people. And so that's why, that would be, I think, one of the reasons why, if not the primary reason, as to why, in male-dominated spheres or environments, a certain element of toxicity can be cultivated, as opposed to that's less likely to happen in a more female-dominated environment. I'm not saying that there a toxic woman or a toxic femme-presenting person doesn't exist. Absolutely, there are totally toxic women out there that have toxic attitudes, discriminatory attitudes, bigoted views, for sure. I'm just saying, on the whole, like, it is generally less problematic to have a field or a sphere dominated by women than it is to have a field dominated by men.
But when there is no pattern, this is a terrible strategy. Even when there are patterns, you need timely feedback in order to learn them. And YouTube knows this, which is why within the first hour after posting a video, they tell you how its performance compares to your last 10 videos. There's even confetti fireworks when the video is number 1. I know it seems like a silly thing, but you have no idea how powerful a reward this is and how much YouTuber effort is spent chasing this supercharged dopamine hit. To understand the difference between immediate and delayed feedback, psychologist Daniel Kahneman contrasts the experiences of anesthesiologists and radiologists. Anesthesiologists work alongside the patient and get feedback straight away. Is the patient unconscious with stable vital signs? With this immediate feedback, it's easier for them to learn the regularities of their environment. Radiologists, on the other hand, don't get rapid feedback on their diagnoses, if they get it at all. This makes it much harder for them to improve. Radiologists typically correctly diagnose breast cancer from x-rays just 70% of the time. Delayed feedback also seems to be a problem for college admissions officers and recruitment specialists. After admitting someone to college or hiring someone at a big company, you may never, or only much later, find out how they did. This makes it harder to recognize the patterns in ideal candidates. In one study, Richard Melton tried to predict the grades of freshmen at the end of their first year of college. A set of 14 counselors interviewed each student for 45 minutes to an hour. They also had access to high school grades, several aptitude tests, and a 4-page personal statement. For comparison, Melton created an algorithm that used as input only a fraction of the information, just high school grades and one aptitude test. Nevertheless, the formula was more accurate than 11 of the 14 counselors. Melton's study was reported alongside over a dozen similar results across a variety of other domains, from predicting who would violate parole to who'd succeed in pilot training. If you've ever been denied admission to an educational institution or turned down for a job, it feels like an expert has considered your potential and decided that you don't have what it takes to succeed. I was rejected twice from film school and twice from a drama program, so it's comforting to know that the gatekeepers at these institutions aren't great predictors of future success. So if you're in a valid environment and you get repeated experience with the same events, with clear, timely feedback for each attempt, will you definitely become an expert in 10,000 hours or so? The answer, unfortunately, is no, because most of us want to be comfortable. For a lot of tasks in life, we can become competent in a fairly short period of time. Take driving a car, for example. Initially, it's pretty challenging. It takes up all of System 2, but after 50 hours or so, it becomes automatic. System 1 takes over and you can do it without much conscious thought. After that, more time spent driving doesn't improve performance. If you wanted to keep improving, you would have to try driving in challenging situations, like new terrain, higher speeds, or in difficult weather. Now, I have played guitar for 25 years, but I'm not an expert because I usually play the same songs. It's easier and more fun. But in order to learn, you have to be practicing at the edge of your ability, pushing beyond your comfort zone. You have to use a lot of concentration and methodically, repeatedly attempt things you aren't good at.
You can practice everything exactly as it is and exactly as it's written, but at just such a speed that you have to think about and know exactly where you are and what your fingers are doing and what it feels like.
This is known as deliberate practice, and in many areas, professionals don't engage in deliberate practice, so their performance doesn't improve. In fact, sometimes it declines. If you're experiencing chest pain and you walk into a hospital, would you rather the doctor is a recent graduate or someone with 20 years experience? Researchers have found that diagnostic skills of medical students increase with their time in medical school, which makes sense. The more cases you've seen with feedback, the better you are at spotting patterns. But this only works up to a point. When it comes to rare diseases of the heart or lungs, doctors with 20 years experience were actually worse at diagnosing them than recent graduates. And that's because they haven't thought about those rare diseases in a long time, so they're less able to recognize the symptoms. Only after a refresher course could the doctors accurately diagnose these diseases. And you can see the same effect in chess. The best predictor of skill level is not the number of games or tournaments played, but the number of hours dedicated to serious, solitary study. Players spend thousands of hours alone learning chess theory, studying their own games and those of others, and they play through compositions, which are puzzles designed to help you recognize tactical patterns. In chess, as in other areas, it can be challenging to force yourself to practice deliberately. And this is why coaches and teachers are so valuable. They can recognize your weaknesses and assign tasks to address them. To become an expert, you have to practice for thousands of hours in the uncomfortable zone, attempting the things you can't do quite yet. True expertise is amazing to watch. To me, it looks like magic, but it isn't. At its core, expertise is recognition, and recognition comes from the incredible amount of highly structured information stored in long-term memory. To build that memory requires 4 things: a valid environment, many repetitions, timely feedback, and thousands of hours of deliberate practice. When those criteria are met, human performance is astonishing. And when it's not, you get people we think of as experts who actually aren't. If you want to become a STEM expert, you have to actively interact with problems. And that's what you can...
I think that's a pretty good video. I don't disagree with it at all. Basically, yeah. If you want to become an expert in anything, you gotta practice at the bleeding edge of your comfort zone and constantly challenge yourself.
We're running out of land.
And even then, like, who proclaims someone as an expert? At least in science, you are deemed an expert when some other committee says, hey, you've graduated, here's your piece of paper. And now you're an expert in this very niche field.
To produce food.
Oh, an oily olive, welcome. No, I did not switch fields. Not yet, at least. I did share at the beginning of the stream that I might be getting a new job. Maybe. I might be leaving my biotech startup company. For basically a writing position at another company.
Good news, we can get our food from somewhere else. Farming fish has been the fastest growing food.
Oh, and you mean John Campbell, right? I'm pretty much, ignoring Dr. John Campbell. I do think he's a fraud. And I have no problem saying that. Not a fan.
Production method in the world, but it comes with problems of its own.
Diseases, nutrient pollution, and overfishing, to just name a few.
So let's go out there and see if we can fix this. How can we farm the oceans sustainably?
Yeah, I eventually figured that out. John Campbell, big fraud.
To find that out, I came here, Ireland, more specifically to the salmon research site of the Marine Institute. It's specifically developed to farm salmon more sustainably, around 20,000 of them per year. At the same time, it produces 3 tons of seaweed and hundreds of different shellfish. I'll tell you why later. First, let's meet the people who run the show. Frank Kane, Joanne.
Well, I would say certainly, as far as I could tell, Dr. John Campbell, they're an expert when it comes to nursing or nursing education. Since that's what allows them to use the title of doctor, not a medical doctor, but like a PhD in nursing education or something to that effect, or like a PhD in nursing. And so I have no problem saying that he is probably an expert in that. But is he an expert on, the SARS-CoV-2 or monkeypox, which I think is what he's reporting on now? I think for me, the answer is clearly no. I don't think anybody should be going to him for that type of information. And it's actually kind of sad to me that he's gotten popular from presenting this information when I can tell, I don't even consider myself an expert on SARS-CoV-2 or monkeypox, and I can tell that he's not an expert in these things, and he's doing a disservice to science in my mind. I think we should optimize our traditional land farm. I don't disagree with you. Especially, I think we should optimize our traditional land for plant-based foods and agriculture. For here, I'm an advocate of, at the very least, plant-based diets. I had a conversation with the juggling vegan YouTuber a few weeks ago. The VOD is now up on the YouTube channel because I do strive to be vegan.
Casserly and Neil Rain.
So how bad are the waves out there? I might get seasick or
No,
not?
no. Today we have a good one. We have quite a light breeze, so it's all fine. We have a little bit of sunshine. It won't be strong, but no waves. So hopefully we'll have a nice day out there.
Okay. These are funky trousers.
The first thing I want to look into is the emissions associated with farming fish.
Fish farms impact the climate in 2 ways. You need to feed the fish, obviously, and you also need the energy to run an operation like this.
Then there are variables
I don't think I know Dr. Malcolm. If you want to send a video of Dr. Malcolm, be happy to check them out, but I don't think I know who that is.
depending on how, where, and what type of fish you farm. It can be in a bay area or back on land with closed-loop tanks like these.
This is surprisingly stable.
The tiny food pellets this machine shoots into the pond are responsible for more than 70% of farm fish emissions, and that's because of what's in them.
It smells quite
fishy.
Yeah. And what's in this?
So this is made from mainly fish meal with cereals and other fish oils. Fish need high quality marine fish products within their food to supply them with omega-3 fats, which they need for growth and health and their immune system. So the primary product going into fish food would be fish meal, so which would be either fish caught at sea or the waste products in fish factories.
And catching fish means emissions. You need to burn fuel to power the fishing boats, and you need to freeze or cool the fish once it's caught.
But there are alternatives. For example, here they fed the salmon with fish waste instead of newly caught fish in the feed. And we do need these alternatives because the small fish that are in this feed are not available at large quantities in the sea anymore.
Other ideas for low emissions fish feed are algae or insects. More commonly, soy is used as a substitute these days, but that's not...
Or just stop farming the fish. That would be my solution. I definitely think, we should not be consuming seafood.
Without its problems.
A lot of it is actually coming from places like America, China, but more controversially in Latin America.
This is Alex One. He has been researching fish feed for years.
Where large areas of forests are being removed to grow soy, and this is being transported across the different parts of the world, and this actually adds a lot of carbon emissions and also have a significant impact on nutrient runoff and social impact as well.
And then you can look at how efficiently you're using the feed. There's the so-called feed conversion rate, short FCR, to measure that. It determines how much feed is converted into live weight by animals. Farm fish are pretty good at this. Their FCR is between 1 and 2.4. For pork, it's up to 5. And for beef, it's up to 10. So farm fish need less feed to gain weight.
And that's because they are cold-blooded, so they don't need as much energy as we humans, for example, to run their overall system. So they can devote more of this feed to growing.
Getting a low FCR has to do a lot with finding the perfect feed composition. This can reduce the fish's environmental impact by as much as 24%. Along with the feed, there's another big factor: the emissions produced from the industry's energy consumption. And this really depends on where and
if we embrace this kind of farming, it will inevitably become corporatized as much as any other agriculture path. I also, yeah, totally. As I've sort of shared before, like definitely, microplastics are present in canned fish, and it seems like the evidence points to like basically seafood as being like one of the highest containing one of the highest concentrations of microplastics. And so like, yeah, if you want to minimize your microplastics eating, I don't want to be eating seafood. It's Wilson, not, nope, Dr. Wilson also does not sound familiar. But yeah, if you want to share a video, by all means.
How you grow your fish. Let's look at salmon, for example. It's bred in tanks on land, or like here, in open net pens in the ocean.
Like, these are awful. Without getting into too much detail, or like, giving away confidential information. But as part of my job for my biotech startup, I was tasked with, basically becoming an expert on aquaculture and aquaculture farming, because we had a client, or we have a client that's like interested in using our device to address a problem in aquaculture, and the problem is not small, and the problem is actually kind of disgusting. But that's as much as I can say. I imagine the highest or second highest plastic content is probably pork. They literally feed pigs. It's definitely probably, it is definitely another animal. I think it'd probably be a competition between pork, well, the standard pork, chicken, or cow. But I think chicken is up there as well.
Okay, the world's most tiniest windmill is here.
Yeah, and it works very well.
So how much energy do you need to supply this whole operation?
Okay, so here we have a small open pen fish farm system. So the requirements for energy on the site are relatively low. We do have a monitoring buoy over here which is solar powered for the monitoring and the communication. We have a small wind energy and we have solar panels on top of this hut. So that's a power source that charges batteries for us to run the feeders and the small structures on site, and then we have a large generator here if we have to pump large volumes of water. From a sea site, this would be a relatively low energy cost one. Generally the open pen system doesn't require the same amount of energy. Recirculating systems, on land systems would have a higher demand because they have to pump water around and heat water.
The difference is actually huge. A US study compared the impacts, and if you keep salmon in tanks on land, your greenhouse gas emissions double. Farmed in open net pens, you're looking at 3.4 kilograms of CO2 per kilogram of salmon, and bred in tanks, you're stuck with 7 kilos.
There is one effective way to get rid of these emissions. Use renewable energies. If you use about 90% renewables, you can cut the emissions.
Another fantastic question. So, my thoughts on nuclear energy, appear to have like evolved over time, based off of, new information. And I will plead, I have no problem pleading ignorance on nuclear energy, as in that's definitely not my area of expertise. And actually, I have thought about reaching out to a nuclear energy expert in the Twitch space to, basically educate me on nuclear energy, because it's something that I want to know more about. Because like for the longest time, okay, I was under the impression that like yes, nuclear energy is a form of clean energy, in the sense that, in the actual production of energy, it's actually quite good. Oh, no problem. Hope you have a safe ride, Oily Olive. Always a pleasure. And so, yeah, when it comes to just producing energy, nuclear energy is like pretty clean. But in my mind, at least as it was sort of taught to me, that there was this issue of nuclear waste. As in, like, what do we do with it? This is like radioactive, decaying matter that we have to contend with. And my understanding is like, the solution was just like to bury it, deep in underground, or like just store it in like vats, store it deep underwater, or like tanks or whatever. But, apparently, okay, this seems to be less of the case, and that, nuclear energy is actually, much cleaner, and that, this radioactive waste is much less of an issue than I thought. And, it seems like the recurring theme of like, if you are actually in favor of, green energy or clean energy, then you should be pro-nuclear. And so it's like, that's sort of where I'm at now. But again, I definitely want to inform myself better on it before, holding firm in my stance. And actually, I will probably look to have a conversation on stream with a fellow Twitch streamer who advocates for nuclear, and it seems like they are a nuclear energy expert, which is why they advocate for it. I think they go by Soyaddle on Twitch. I'm probably going to reach out to them and have a conversation on stream with them soon, and probably field some questions from chat if there are any. Cause less death per watt produced. Yeah, that makes sense to me.
of land-based systems toward the emissions of an open net pen system like here.
But the world average of renewable energy in our mix is currently at 29%, so nowhere near that. And sadly, the problems don't end there.
If you have a lot of fish in one place, you also have a lot of in one place. And when that gets too much, things get ugly.
Too much fish poop in the water can in the worst case lead to an algae bloom that can eradicate all fish in your farm and tons of sea life on top. The fish basically suffocate underwater. Their excrements work like a fertilizer, so algae grow like crazy and block out the sun, which kills many other underwater plants. This means less oxygen is produced, and the bacteria decomposing the dead plants take up what little is left. In the end, the complete ecosystem collapses. In 2021, along the Chilean coast, 4,200 tons of salmon perished like this.
For these 20,000 fish we got here, how much fish are we actually talking about?
So based on the literature values that we have, the waste from the fish would be at about 20% of, say, maybe what they're fed per day. So on our site, with our amount of fish, that works out at about 3 kilos.
That's about one.
Any climate change studies recently or lately? So do you account for the CO2 consumption of plants? Well, to answer your question, 1 second. I mean, I absolutely do have research, and I've read quite a bit of research in regards to climate change, but not really devoted to your specific question of CO2 consumption of plants. But I'm more than happy to look into it. I am mostly focused on like how to basically convince as many people as possible that climate change is a thing. It's a problem that we need to address. It's a global problem that, you know, the world needs to address. Which is why it's like, yeah, you know, I clearly read climate change research because it's published in Nature Climate Change. But, you know, it's articles about, you know, what type of groups are basically, you know, resisting accepting climate change as like a real thing we have to address. And that, you know, understanding and managing connecting extreme weather events, and also, you know, views on climate change, like how do we get, again, certain groups or identities to recognize that it's a problem. I also, you know, have research in terms of like how can we as a consumer, if we're interested in you know, reducing our impact on the environment or reducing our contribution to climate change. Well, one of the ways that we can do that is through, you know, dietary choices, which is why I advocate for, at the very least, a plant-based diet, or at the very least, you know, eating more plants, incorporating more plants in your diet, because the evidence is there robustly that, like, without question, eating plants has much less of a negative impact on the environment. Well, I hope, you know, I hope so, Bob Marley. I really do. But at the end of the day, like, I could be wrong, but I don't really think libertarians care about climate change. I don't. But maybe I'm just too much in the Twitch world. Because of all the libertarians that I know, they're like, yeah, whatever. You know, the free market will come up with a solution. You know, it's just a matter of time. Like, I don't think that's happening. Because I keep reading that CO2 molecules can remain in the atmosphere for centuries. Yet I've also read that it gets absorbed by plants almost immediately before it gets high enough. So, okay, if you're specifically referring to CO2 molecules, are you sure you read that specifically? I'd be very curious where or like what article you got that from. Because like, yes, many things get released into the atmosphere. CO2, methane, other gases, and molecules, and they all have their kind of like own cycle. And even then, CO2, I think there's a little bit of, CO2 is a greenhouse gas emission, but it may not be the most problematic. Just because, like, yeah, there appears to be a cycle to CO2. It's a long cycle, but in terms of like its potency, it seems like maybe methane is more potent as a greenhouse gas than CO2. Both are problematic, but it might just be methane that is, whatever, the most problematic. I will build my own personal biodome, says. Well, I'm hoping it turns out better than the movie Biodome, because that was an awful movie. It was great back in the 90s. I think libertarians have very strong political incentives not to accept it as a problem because their theory relies on it. Yeah, I think that's the problem. I think they're just so free market oriented that like if any solution to a problem requires going outside of the free market, like I don't think they can recognize it, or I don't think they can even acknowledge it. That's my suspicion. Water vapor. I will plead ignorance on that, but I'd be curious to know as to why are you saying that, or where are you getting this information from?
Ton per year, and when you overfeed the salmon, it gets even worse. But the researchers here have an idea how to fix this. The magic word is polyculture, so growing different varieties of seafood alongside each other.
So what's the kind of service that these scallops provide for this overall system?
So we've chosen scallops. We've also have oysters as part of our shellfish level, and they're what we would call extractive species. So they would extract all of the particulate matter out of the water column. And how they do that is they have what we would call a beard, and it's within the shell here, and that filters the water and takes the particulate matter out of it, and that's their food source.
So they basically eat the shit?
Basically, yeah, but they would eat, say, the other particulate matter that's available in the water column too. So any zooplankton, maybe bits of detritus, or broken up bits of organic matter is all available to them to eat as well.
And because of that, it's important where the scallops are placed, because you want the current to bring the fish poop to your shellfish and not elsewhere. And the good thing environmentally is the scallops don't require any additional feed, so you just hang them out in the water and wait for them to grow. The same goes for the next seafood that helps clean up the salmon's mess.
That's quite a bit of seaweed!
More specifically, sugar kelp.
You can eat it?
Yeah, if you wanted to take a little bit to try.
Okay. Yeah, it smells salty. But it's actually very much like salad.
Yeah.
And nice when you forgot your lunch, right?
Yeah.
It grows along the main line that's just put in the water. It almost
doesn't look too appetizing to me.
Looks like a root system, from your regular houseplants. And it grows really, really quickly.
So we're at about 8 kilos a metre here.
8 kilos a metre?
Yeah, this is getting to really more of the higher growth season for the time of the year. So it really prefers the colder waters. It grows throughout the winter. And around now it's putting on maybe almost kind of 1 kilo a week in terms of growth rate.
1 kilo a week? That's insane.
Yeah. So it's really fast growing plant material.
In addition to the shellfish feeding on the small poop particles from the fish, the seaweed absorbs anything that's already dissolved in the water, like nitrogen and phosphorus that can also lead to algae blooms. It's an on-top extraction system that also produces more food, seaweed. Growing all these things at the same time makes the whole farm more sustainable and productive. But to what extent can the other species really offset the impact of fish farming?
So probably averaged out overall, including maybe the filtration rates of some of the shellfish, about 20% of what we're producing here in terms of salmon is being offset by the extractive species. And that's only with, there's about 400 to 500 metres of long line out at the minute, and only about, say, 600 to 900 shellfish, so very, very small quantities.
A polyculture farm like this can take care of one problem, nutrient pollution. A big one, yes, but other issues such as the spreading of diseases amongst farmed fish or chemical pollution with antibiotics remain. And that's...
Yeah, so, again, without giving too much away, one of the huge issues in these, like, open water, like, fish farms, where it's just, like, a bunch of fish in a net in the water, is, like, infection. Or like disease spread, because like you are literally keeping whatever hundreds or thousands of fish just like confined this small cage, and like if one fish gets infected, well then it just like runs its course throughout the entire population, and you just have like massive die-offs of like thousands, tens of thousands of fish.
Speaks to the crux of...
And so like one of the huge issues or challenges in these, you know, aquaculture farms, which they market as like, oh, this is like this is more environmentally friendly, which sure, maybe technically it is, but it's at the cost of like, you know, hundreds of thousands of fish, where it's like, again, the huge challenge is like, how do we manage infection? How do we prevent these fish from getting infected, or just one fish getting infected and then it just spreads to the rest of the fish?
Of the matter.
We can reduce the fish's impact, but we cannot eliminate it. Alternative feeds, polyculture, they don't solve the problem. If we want to do that, we need to get rid of the fish. I know it's super sad, but I mean, the seaweed didn't taste too bad after all.
If you enjoyed me eating seaweed grown on fish poop, please subscribe to our channel. We post new videos every Friday.
Oh, thank you so much for sending a link. Oily Olive. Always pleasure. Assuming this is Dr.
Hey, I'm Dr.
Oh, okay. This person seems familiar. I think I've seen them appear on Twitch. Do you think there are any positive effects of climate change? I've heard that specifically CO2 in high enough volumes can actually produce a cooling effect globally. Not to mention that a higher carbon dioxide content in the atmosphere could result in faster plant growth. Well, so yeah, like, yes, that is true in terms of like, and I think we're even, I think there's evidence showing that the higher CO2 is affecting plants and is actually like accelerating plant growth, and that, you know, this high CO2 content is good for the plants. But you know, what's good for the plants doesn't mean it's good for the planet, and that there are potentially still negative side effects associated with that, of like depleting the soil, if the plants are growing too much, or, you know, too accelerated, you run the risk of like, you know, depleting your soil of nutrients, and kind of like potentially maybe like creating a desert somewhere down the line, where like just that soil is no longer like usable land for growing plants. Like plants grow at such an accelerated rate. But aside, yeah, I don't think there's any real positive effects of climate change, as far as I can tell, other than, you know, maybe some colder places see more warmer weather, or like, you know, during the winter months here in Quebec, like we have more warm days than we used to have like 10-20 years ago, and that it seems like the winters are getting more and more mild, but that's at the expense of like pretty extreme heat waves during the summer.
Wilson. I'm a PhD molecular biologist, and welcome to another COVID debunking video. This week, I'm looking at a very stupid claim that just won't die. It's the idea that COVID vaccines...
I mean, if he's a microbiologist on their PhD, then probably they're almost certainly way better qualified than Dr. John Campbell.
Change your DNA. They don't, but that hasn't stopped anti-vaxxers from lying any way they can about it to try to make it look like that's the case. And now it's going viral again thanks to people like Peter McCullough and Simone Gold, two anti-vaccine liars that are just ridiculous. So in this video, I'm going to be going over what needs to be demonstrated in order to prove that the sequence in the mRNA COVID vaccines has been integrated into a human genome, and then I'm going to talk about why the paper that Peter McCullough and Simone Gold are referencing is totally garbage and how they're lying about it. So let's get started. There are a very specific set of experiments that you can choose from in order to demonstrate that the COVID mRNA vaccine sequence has been integrated into a host genome. Molecular biologists know how to do this. We have to do it regularly when we are manipulating the genomes of the cells that we work with. We have to make sure that any constructs that we're adding into the genome have actually been added into the genome. This includes specific PCR-based sequencing techniques in order to demonstrate that the sequence that you're trying to integrate is actually in the host genome by showing that it is flanked by host genome sequences. You could also do a whole genome sequence of the cell lines you're working with, or you could do something like a southern blot. The point to all of this is that there are a specific set of experiments that need to be done in order to demonstrate this. And the paper that Peter McCullough, Simone Gold, and others are referencing does not do those experiments. It does not show integration of the COVID mRNA sequence in a host cell. It doesn't show it at all. And if you hear any future claims of this happening, you should be asking the question, does the paper actually show integration into the host genome with any of these specific experiments? If it doesn't do that, you can dismiss it outright. But let's go into a little more detail about what these anti-vaxxers are actually saying and what the paper actually shows.
The study that just came out of Sweden that is just so alarming.
The news is buzzing out of Lund University, Malmö, Sweden. Marcus Alden is the first author. The first demonstration in a human hepatic or liver cell line that the Pfizer vaccine, in fact, reverse transcribes and installs DNA into the human genome.
Actually, the paper only shows that first part. It does not show integration or installation, as Peter says, into the genome. So let's talk about this a little more in detail. The active material in
If there was a great greening effect, it's thought that places like the Sahara would become green again. Well, let's just say I'll believe it when I see it. I'm not discounting that, but I suspect it would take more than that to make the Sahara green again.
In the COVID mRNA vaccines is the mRNA. mRNA is made from DNA in a process called transcription. This requires a set of enzymes to read the DNA and turn it into a different molecule, which is RNA. The information in that RNA is then read by other cellular machinery in order to make proteins, which do a lot of the work in the cells. The mRNA itself is really transient. It doesn't last very long. It's only meant to be used as a copy of the information that is.
No, absolutely not. That is not the function of our mRNA in your body. Maybe I can show you. One second. I will attempt to show you. Give me a moment. I'll let the video play as I search.
Is stored more permanently in DNA. So mRNA does not get replicated like DNA does. It does not get passed down from cell to cell like DNA does. It's just a temporary copy of the information. So in order for the information in the mRNA vaccines to become a permanent part of a host genome, it has to first be reverse transcribed. That means it has to be turned from mRNA back into DNA. And This takes work. It takes the work of enzymes to do this specific job. Once it gets reverse transcribed back into DNA, it then takes another set of enzymes to incorporate the DNA into a host genome. And the DNA usually has to have some specific characteristics that will help it get integrated into specific spots in the host genome. This paper that McCullough is referencing here only shows that first step of reverse transcription, and the researchers actually forced it to happen in unnatural conditions. Let me explain. Human cells don't have much reverse transcriptase activity. Reverse transcriptase just refers to enzymes that can turn mRNA back into DNA. Human cells don't do this much. But one of the enzymes that the human genome does encode that can do this is called line 1. Line 1 is a reverse transcriptase that usually just replicates itself. It has an affinity for its own
Okay. Great. Right from a textbook. A molecular biology textbook. I was taught in biology that is used by viruses to change your cells to produce viral cells and is used by your cell to communicate with its membrane or something. So totally. Viruses that contain RNA is a thing. Absolutely. And viruses can use and do use mRNA. HIV is one of them. SARS CoV-2, I believe, if I remember correctly, is one of them. But the function of mRNA in a human organism, okay, it's like it's part of the process of like making the things that your body needs, or it's the pathway to get protein. Second, well, it's highlighted here. The pathway from DNA to protein, as in the end goal, okay, of the genetic information in your body, of your useful genetic information in your body, is to make proteins. That is what it's concerned with. Proteins are like, the actors that, carry out virtually all of the biological processes and reactions in your body. Proteins make up the structure of you as a human. Proteins carry out virtually all of the chemical reactions that take place in your body. And you are constantly making proteins in your body. And so RNA, that is what I guess you could say is the stepping stone. As in the DNA that contains your genes, okay? And that contains, the information of basically how to assemble the protein. But the DNA is not read directly because the DNA basically can't be read directly. One of the primary, yes, absolutely. In this context, RNA and mRNA are the same thing because the RNA that the vast majority of the RNA that gets made in your body is messenger RNA. This is explicitly mRNA. It will likely be contained here somewhere. Second. Well, yeah, it's even in the name of the chapter. How cells read the genome from DNA to protein. 1 second. But yeah, it is synonymous at this point. And so RNA or mRNA is just like the intermediate point. As they sort of highlight here, the DNA gets transcribed into RNA, and then the RNA gets translated into protein. So it's like this two-step process: DNA to RNA, RNA to protein, and protein or proteins are what matter, or like pretty much what matter the most when it comes to the important actors in your body. And at least in a human organism, okay, it is virtually, well, it's impossible, okay, to go backwards from protein to RNA. As in, once the protein is made, you basically cannot. There's a reason why this arrow is in one direction. You basically can't go backwards, or at least without some sort of like external intervention by some sort of tech. Generally speaking, your body doesn't have the means to go backwards in this process. As in, it's like unidirectional. You go from DNA to RNA, and then you go from RNA to protein. And what I can say with absolute certainty is that you cannot go from protein to RNA, and that in another context, okay, some organisms can go from RNA to DNA, but generally speaking, on the whole, RNA in a human organism can't go back to DNA. Yeah. So you are posing an excellent question, and the answer is contained basically in this diagram, because I think everyone knows now that the mRNA vaccines, okay, they code for the spike protein, okay, and so when those vaccines that contain mRNA, like they're just injected, and it's literally just an injection of this RNA that gives your body the instructions to make the spike protein. Like it's literally just an injection of this, but the code is the code for the spike protein. That's it. Like, you are literally giving your body the instruction manual to make the spike protein, and therefore it makes it on its own. That's how the mRNA vaccines work. The mRNA vaccines are not giving DNA. The spike protein is part of, the spike protein, that is a specific portion of the SARS-CoV-2 virus, as in it's one of the outer proteins. That's why it's called the spike protein, because it appears to kind of like protrude out from the virus. But the complement system, that's different. The complement system in a human, that is part of your immune system. Like the complement, like your body's complement response, that is a specific immune response. And so those are two separate things. Like the spike protein is part of the SARS-CoV-2 virus, but the complement system, that is explicitly part of your immune system. I absolutely remember that from my immunology courses. It's a pretty complex system. Right, the complement system. Where it's part of the immune system, and it's a very complicated cascade of events. Well, as outlined here by this diagram. I had to memorize this at one point, but no longer really in my brain. Where it's the complement system, provided you are, a whatever, typically healthy human being, that's part of like your inflammatory pathway to generate inflammation. Generally speaking, healthy inflammation. So the vaccine gives your body mRNA, and this mRNA can, that's one way to frame it. Like, I wouldn't say you are wrong. It's more like, so what happens is that The vaccine gives your body the mRNA, so the instructions to make the spike protein. Okay, so it gives the mRNA to make the spike protein. Okay, as in, like, you get the injection of the mRNA spike protein, and then your body translates it itself, because we have the machinery to translate mRNA, and therefore we make the spike protein. But then from there, it's not necessarily how to destroy COVID. What happens is, even though, okay, your body, when injected with the mRNA vaccine and it makes the spike protein, even though your body is making the spike protein, your body still recognizes it, that it is a foreign protein. Okay? Your body has pretty sophisticated kind of like defense systems or recognition systems. As in, it will generally know, assuming you're a typically healthy human being, it will know if you are making, even if you're making it yourself, a foreign protein. And so it will recognize it as a foreign protein, and then your body will mount an immune response to it. And part of that immune response is generating an antibody response. As in, you will generate antibodies to the spike protein, to the SARS-CoV-2 spike protein, where, like, you know, it will act as essentially a physical blockade acted against the spike protein that prevents it, okay, from infecting your cells. 1 second. I will attempt to sort of make my point clear. Second. So, the way in which SARS-CoV-2, like, actually gets into your cells, your respiratory cells, or largely your respiratory cells, is through the ACE2 receptor. As in, certain cells in your body, they express the ACE2 receptor. Okay, and it's specifically through the interaction between the ACE2 receptor and the spike protein that allows the SARS-CoV-2 virus to get into your cells. Okay, but if you've been vaccinated with an mRNA vaccine, and now you have antibodies against the spike protein, well, what would happen is, if you happen to be exposed to the SARS-CoV-2 virus, and you have antibodies present in your system against the spike protein, then those antibodies, okay, will then bind to the spike protein. And they will essentially sort of cover or block the spike protein. And if the spike protein is blocked, or like covered in, you know, antibodies, then that will prevent it from binding to the ACE2 receptor. As in, it pretty much, it acts as a physical block. If the spike protein on SARS-CoV-2 is covered in antibodies, then it cannot physically interact with the ACE2 receptor. And that's essentially one of the primary mechanisms in which the vaccines protect you against infection. Where it's like, they physically act as a block from SARS-CoV-2 from gaining entry or binding to your ACE2 receptors. Hopefully that clarifies it. If not, let me know. Could someone, not saying that someone does, put information in this vaccine that would be detrimental to your body? As an example, let's imagine it could cause your cells to become cancerous. Could someone, that would be, no, that would be exceptionally difficult. This is one of the reasons why, the mRNA vaccines are generally considered quite safe. Not perfectly safe, but quite safe. Is because, it's generally only useful to you in the context of like, getting your body to, mount a defense against a foreign protein. Because, like, I could be wrong, because I'm certainly not an mRNA vaccine expert, but I'm quite familiar with the technology and just the overall biological processes. And I think it would be exceedingly difficult to somehow design all you can do. You are limited by what can be translated. Okay, as in, assuming you're using an mRNA vaccine, then you're limited by just giving instructions as to what can be translated. And you're either translating a foreign protein or a native protein. If you're translating a foreign protein, as in the case of the spike protein, well then your body will mount an immune defense, and eventually your body will clear it. And we bank on that happening with the mRNA vaccines currently. Like, we don't want the spike protein, just stuck around in your body for the rest of your life. It's a temporary thing. Like, your body mounts an immune response to it. And so, you're either generating a foreign protein or a native protein, as in a protein that you already make. I mean, if it's a protein that you already make, well, then who cares? I can't imagine that there would be any real negative effects from that. But I could be wrong. I think that would be a huge challenge, and I don't see why anybody would be able to get your body to make something detrimental. Because your body is actually pretty good, assuming you're a typically healthy human. Your body's pretty good at recognizing things that don't belong, like the spike protein. Where? Oh, yes.
Sequence. But if it is present in high amounts, it can have off-target effects. So what the researchers did in this awful paper is they used a human liver cancer cell line, one that overexpresses line 1. It has way more copies of line 1 present than normal healthy cells. They then gave, in the cell culture, a large dose of mRNA that is normally put in the COVID vaccines. So we have a lot of mRNA, more than what we would have in a vaccine, and a lot of line 1, way more than what we would have in our normal cells. And the researchers, unsurprisingly, measure reverse transcription of the mRNA that was given to these cells. And that is all that the paper shows. It does not show incorporation of the reverse transcribed.
Well, you'd be hard pressed to get me to say that anything's impossible, but I would say it's approaching impossible. Where it's on the order of so difficult that it's quite close to impossible. But I would never say it can't be done, because who knows? Does DNA change in general? This is a fantastic question. The answer depends on what you mean by change. But basically, does your DNA stay exactly the same throughout your entire lifetime? The answer is no. Almost certainly not.
DNA into the host genome. Not at all. The experiments aren't even there to look for it. But I have a suspicion that the authors actually did those experiments to look for integration, didn't find any integration, and just didn't care to report it. They just published their paper as it is, knowing how it would be received by anti-vaxxers, and they didn't care. And the predatory journal that it was published in, MDPI, likely gave them little resistance at all when they were publishing. And yet, McCullough and his anti-vaccine grifter friends are all parading this paper as if it shows that we are all genetically modified organisms now or something. It is absolutely ridiculous. I'm not even kidding. They're actually claiming that. Here, look.
So one of the fun things about the last couple of years being a doctor and lawyer is this is a very interesting opportunity, and I think we're going to be bringing another lawsuit because there's actually federal law that you cannot discriminate against people for genetic discriminations. I forgot the acronym. I think it's GINA, G-I-N-A. GINA, right? So this now opens the door to saying that if you're not allowing somebody in who chose not to get the shot, that you're actually engaging in genetic discrimination. So I think there's this Sweden study, I think, opens the door to a new type of lawsuit.
Yeah, no, that is just cuckoo bananas. Not only does the paper not even remotely show that, but that lawsuit is absolutely ridiculous and would get thrown out of court. It's just a waste of time. But it's not surprising that this is coming from Simone Gold, who just got out of jail.
If you're pregnant and you have this done, can that then affect your baby?
Yes, see, this is the alarming finding. The CDC says on its website very explicitly, this will not change your DNA.
Because it doesn't. It doesn't change your DNA.
The paper that came out of Sweden. Now, there'll be many more to confirm it. And the steps are to A, confirm it, to confirm that the entire code is installed, and then to actually confirm that it's expressed, meaning that the spike.
Yeah, I'm not sure why she jail. I'm definitely, like, I'm familiar with Peter McCullough, but any of these other people, these women, don't know them.
Protein now is continuously expressed from human cells. But because the lipid nanoparticles are taken up everywhere, that means somatic cells in your organs, but also your gametocytes, actually the cells that actually are the sperm and the egg. If they are carrying it, that indeed means that, in fact, it could be passed to the daughter cells and
So that could be
embryo.
And this is the whole point of their lies. They are trying to muster up fear about the vaccines. They're making these absolutely ridiculous statements that have no bearing in the scientific literature. And they're parading them as truth, all to scare people in order to not take vaccines. It's blatant fear-mongering for their own personal political gains. So just to recap, there are two major steps that a cell needs to do in order to integrate an RNA sequence into its genome as DNA. There are other steps in between, but for simplicity's sake, I'm just focusing on these two major steps. The first step is reverse transcription. The mRNA has to be turned back into DNA. This is not an easy task, and the researchers had to really force it to happen in their cell culture conditions that they were working with. This would not normally happen in your body after a vaccination. The second and most important step is the actual integration of the DNA into the host genome. This is a tall order for cells. Not only does it require certain enzymes that aren't usually present in human cells, but it also will require some certain characteristics of the DNA itself. It has to have some matching regions flanking its ends that help it actually recombine and integrate into the host genome. None of that is present in the context of COVID mRNA vaccines. Without those components, it can't happen. COVID mRNA vaccines will not change your DNA. The components aren't there. It's not going to happen. You don't need to be afraid of it. And sure enough, we don't see any evidence of this happening in animal models or in humans. From all of the data we've gathered, the mRNA and the spike protein stay mostly within the injection site of the vaccination. And after a few weeks, all those components are faded away and gone because they are transient. They are not being permanently incorporated anywhere. But hey, since when do anti-vaxxers care about actual data? They don't. They never have and never will. They're just going to keep lying about it, but hopefully you can stay informed enough to not fall for their lies and actually listen to the science and the data that scientists work so hard to reproduce. Well, that's going to do it for this week's video. As always, all of the links to
Yeah, I'd say overall pretty good video. And I like their approach. And it seems like they know what they're talking about, which doesn't surprise me since they appear to have a PhD, I think, in microbiology. So thank you so much for sending that, Oily Olive. Much appreciated. Let me catch up. Chat. If it does change DNA, is that a bad thing? Why is changing DNA bad or could be bad? So yeah, if it so even though I think it was quite clear And I want to be clear, it doesn't change your DNA, but hypothetically, if it did, then yes, that is hypothetically not good, because, okay, it might result in you having sort of bad instructions for making a protein. Where it's like, yes, the DNA contains the blueprint, okay, for making the protein, and it's a particular sequence of code, okay? And so if, something changes your DNA, it's not a guarantee, but there is a decent chance that you might be changing the outcome of the protein. And if you change the outcome of the protein, there's potentially negative effects, ranging from mild to severe. Hard to predict exactly what the consequences would be. But generally speaking, once you start altering a protein from its native conditions, you are usually not changing it for good, and more often for the bad. Does every cell in your body have the exact same DNA sequence? If so, how do you have... This is a fantastic question that I'm going to do my best to answer. Give me a moment. Just 1 second, just going through my dozens and dozens, thousands of papers. Right. Okay. So, there's basically 2 questions. Does every cell in your body have the exact same DNA sequence? If so, how do you have different types of cells? So, to answer your first question, does every single cell in your body have the same DNA sequence? The answer is no. Actually, there is more, okay, DNA variation within your body than we previously thought. Which is highlighted by this paper, titled "The Diplogenome Sequence of a Human Individual," where it's like they sequenced, just one person. It was this person, who was basically, the person in charge of the study. So it was a little bit egotistical, where it's like this person, a very famous molecular biologist, pretty much said, hey, I'm going to sequence my genome. And hopefully with that, we're going to my entire genome. And, hopefully we can answer some questions from that. And some of the questions that we got answered is that actually, not every single cell in your body has the exact same DNA sequence. As in, there is, small variations, largely inconsequential. But basically, the DNA sequence throughout your entire body in every single cell, it is not 100% identical. As they sort of highlight here, inclusion of insertion and deletion genetic variation into our estimates of interchromosomal difference reveals that only 99.5% similarity exists between the two chromosomal copies of an individual, and that genetic variation between two individuals is as much as 5 times higher than previously estimated. As in, like, this number was whatever, lower than they expected. As in, like, oh, there's actually more differences than we thought. And so it's like, yeah, we are not perfectly genetically uniform throughout our entire body. And so, to answer your question, your other question of how do you have different types of cells, that's also an excellent question. Where it's like, yeah, if they are all if they all contain the same genetic information, and how come you have, liver cells, kidney cells? And have a lovely evening, Oily Olive. I hope you got some good rest. And I'm sorry to hear that work was rough. I'm hoping it's a better day for you tomorrow. I hope you get some great sleep. And take care of yourself. So then, if basically every cell in your body does actually contain just about the same, you can consider it identical, not perfectly identical, but all your cells essentially have the same genetic information, then how do you get all of the different types of cells that you have? Skin cells, liver, kidney, pancreas, etc., etc. Well, that comes from gene expression, where it's like it's a matter of which genes are turned on in each cell. As in, for a liver cell, the only genes that are being expressed are the ones to, be a liver cell. And then in your kidney, the only genes that are needed for being a kidney cell are being expressed in a kidney cell. You can think of it as like a book with all the various chapters related to the different types of cells, where it's like, you have your giant book that's like all your genetic information, but you have a chapter to the liver, you have a chapter devoted to the kidney, and then in a liver cell, like, then the liver cells, they're only reading the liver chapter of that entire book, and then the kidney cells, they are only reading the kidney chapter of the entire book, the entire genetic information. That's how you have different types of cells. My only question is how accurate the papers are that they base their claims on, and who funds the people writing them. It's well known that the FDA and CDC get a lot of their funding from, and that is a huge problem, and that is a good thing to keep in mind, of where funding is coming from. These are not bad things to keep in mind, and it's pretty much always good to, have a skeptical eye, but also read the science and take it at face value, and also keep it in mind that just because there might be a conflict of interest, or that it might be funded by a pharmaceutical company, that doesn't automatically mean that it's bad research, or that you can't trust its findings, because there are totally examples of studies that have been funded by pharmaceutical companies that have failed. There are well-known stories about that. I think I can dig one up right now. Give me a second. This is a pretty, at least in my mind, pretty notorious case. Yeah. So, this is a drug, etanercept. And you can even be able to see somewhere. Supported by a research grant from Immunex Corporation. Immunex is that's the company that makes the drug etanercept. You could even see here that, you know, they're attempting to be transparent about their conflict of interest. And that William Sanborn is a consultant for Immunex. And they're one of the authors of this study. And yet, despite that, okay, this drug was shown to be ineffective for what they were testing for. In this case, active Crohn's disease. Where you can see it in the conclusion, subcutaneous etanercept at a dose of 25 milligrams twice weekly is safe but not effective for the treatment of patients with moderate to severe Crohn's disease. The dose of etanercept administered in this study is that approved for rheumatoid arthritis. Higher doses or more frequent dosing may be required to attain a response in patients with active Crohn's disease. Where it's like, yeah, this was a study funded by a pharmaceutical company. It's funded by the pharmaceutical company that makes this drug, and yet it's basically negative findings. And essentially, it's based off of this study that the company decided to, like, basically abandon further research on this drug for this condition. It's still used for other conditions, but they were like, hey, this doesn't appear to be useful for Crohn's disease, so let's just forget it. And basically, no real trials occurred after this. It's a massive conflict of interest. The FDA gives the okay on a product, the vaccines, then the governments mandate their citizens to get the vaccine, the companies that sell the vaccine. I don't disagree with you at all. That is problematic. And that, you know, if this was a regular occurrence, and that if this was not, you know, pretty extenuating circumstances under a global pandemic, like, I would have issues with it. But in the context of a global pandemic, you know, when the world is dealing with this virus, I sort of, I can deal with sort of that conflict of interest. And that, you know, it's an exception, not the rule. But let's see. Yeah, I'm going to end the stream. But one last video. One last watch of
This
something
vibrant
scientific. Why spider silk is stronger than steel.
cape and shawl are the largest pieces of cloth to ever be made from spider silk. Over the course of 3 years, it took a team of about 100 people in the highlands of Madagascar to collect the silk, spin the threads, weave the fibers, and embroider the details. In the end, 1.2 million golden orb weaver spiders, each producing 30 to 50 meters of yellow thread, were used to create this masterpiece before being released back into the wild. But why would anyone go to this amount of trouble to make something out of such a strange and difficult to obtain material? Because spider silk has what seems to be almost magical properties. For thousands of years, humans have been
just touching upon sort of this idea of conflict of interest, where it's like, just because a study is funded by a pharmaceutical company, that doesn't automatically mean that, you know, it's bad research or that, you know, you can't trust its findings. Because I was showing an example of like a study that was funded by a pharmaceutical company. Okay, this drug is made by a particular company, and it's shown down here, where, as they say, supported by a research grant from Immunex Corporation. And Immunex makes this drug, etanercept. And so, despite that conflict of interest, or despite the fact that, you know, this pharmaceutical company was funding this research on this drug, they found negative findings, as in not effective. So they abandoned it. And so just trying to get the message across that just because a pharmaceutical company funds certain research does not automatically make it bad. Oh, well, I have no problem choosing another video. There's other science videos that I can choose from, which is this one. Hopefully you don't have a fear of crows.
In the animal kingdom, intelligence has evolved in many different ways in many different creatures. We often think...
Yeah, I did. About this conflict of interest and the FDA and that the mandated vaccines. And my response was basically like, I don't disagree with you in that, you know, if this was a different context, like outside of a global pandemic, like I would be less okay with it. But given the fact that this occurred in a global pandemic, like I was more okay with that situation. And that this is an exception, not the rule.
Think of our fellow primates at the top of the intelligence list, with their ability to use tools like sticks and rocks to access food, and with their ability to recognize themselves in the mirror. Orcas, too, are among the most intelligent, with their unique language skills and orchestrated hunting techniques. And anyone with a dog is constantly surprised at complex feats of social intelligence and emotional sensitivity. Intelligence seems to thrive in animals like this, mammals, often carnivorous ones, that live in tight social groups. But if we survey the world of animals, one of the smartest is one that is perhaps unexpected. An animal that isn't a mammal at all, but a bird, crows. Or more generally, corvids. Corvids are mischievous, playful, and extremely intelligent. They use tools to forage and to solve complex problems. They even drop nuts on a road for cars to crack, and then pick them up with the safety of pedestrian lights. They can also recognize faces, and can hold a grudge against someone who was rude to them for several years. They can mimic sounds that they hear, and scientists have found that their problem-solving skills rival that of a 7-year-old human. How is it that crows are so profoundly intelligent? And how is it that we even measure their intelligence in the first place? Is bird intelligence wired in the same way as mammal intelligence? Or is it something different altogether? The corvid family is also known as the crow family, and includes crows, jays, ravens, rooks, and magpies. The level of intelligence varies between the species, but scientists have found that the New Caledonian crow is the most intelligent. They're to corvids what we are.
I've heard sort of speculation about that, but I don't think there's any, as far as I know, there's no evidence to support that. But I'd be more than happy to look at evidence if someone says that there's evidence to support that.
To the rest of the primates. In 2014, the crows were presented with various tubes filled with water, with a treat floating on top, which they couldn't quite reach. In one trial, when presented with a treat inside a tube of water and a treat sitting on top of sand, the crows knew to drop rocks into the water-filled tube to raise the water level to reach the treat, and ignored the tube with sand. In the next trial, when presented with a single tube of water and objects with various densities, the crows knew to select the sinking objects rather than the floating ones to reach the treat. When presented with 2 tubes of differing water levels, the crows knew to drop objects in the tube with the higher water level to reach the treat faster. These trials demonstrated the crows' abilities in both problem-solving and tool use, and the authors believe that their skill level at deciphering and completing these tasks is similar to 5 to 7-year-old children. However, in 2...
That's pretty kind of evidence could you even get? So, you would... Good question. You would probably have to show that, you know, there's something anomalous in sort of like, at the genetic level, where it's like, you know, based off of genetic information, we're able to like trace back pretty far in terms of like, you know, our ancestors, like pre-human ancestors, and like sort of track the evolution of humans over time from, whatever early apes or primates, Homo sapiens, or like Homo sapiens sapiens. So like, yeah, it would be quite a challenge to like call that into question. And so like you need pretty convincing evidence. But you'd have to show like, oh, there's some sort of like genetic anomaly on like on the pathway of our evolution, that that would be evidence of like, oh, some sort of tampering has occurred. May be extraterrestrial.
2 more challenging tasks the crows didn't perform as well. One required understanding the difference in a wide tube versus a narrow tube, and another was a counterintuitive task, where the tube with the reward was connected to another tube via a hidden U-bend. The crows didn't fully grasp these setups, but in comparison, during tests with human children, only kids around 8 years old were able to consistently solve such tests. And in addition to using tools, New Caledonian crows can build tools. And not just simple ones, but ones with multiple parts. When given objects that were too short to retrieve food from a puzzle box, the crows combined elements by inserting one stick into the hollowed opening of another to make a tool long enough to reach the food. One crow even made 3- and 4-part tools. Compound tool construction like this has only been observed outside humans in a few captive great apes. It's usually only associated with human intelligence. And in addition to this, we now think crows might be better than we ever could have imagined.
We find in the skeletal record that brain skull size increased. I think it was like 40. Well, yeah. So that's where there's a little bit of nuance there. I think because like the 20,000 years, while sure, on a universe timescale, that's not long, and even on the course of evolution, not super long. But something increasing sort of at a somewhat accelerated pace over the course of 20,000 years. That's not. It's hard to argue that that's evidence of some sort of genetic tampering, because that's showing. If you break it down, whatever, what's 43% divided by 20,000? Right? It's like some pretty small number. As in like skull size is changing by 0.002% each year. Increasing. As opposed, like if some sort of extraterrestrial intervention is like tampering with our genetic information, you would probably see some sort of effect that's like an overnight effect, as in like going from like whatever percentage one year that your baseline, and then 43% within a year or so, or like 2 years, where it's like there's this jump, as opposed to like 43% over the course of 20,000 years. Well, that's 0.002% per year, and that's evidence of like small, sure, maybe more accelerated in comparison to other primates, or whatever other organisms, but it's still showing some sort of incremental change over time that's cumulative, and that speaks to something more driven by evolution, or some sort of selection, than some sort of extraterrestrial tampering or manipulation.
At making plans for the future. The ability to take actions now that will give a benefit later is not something that most animals are good at. Delayed gratification and planning are high-order skills. Human children can't do this, and neither can many of the adults that I know. It requires our memory of the past and foresight for future events. Can crows, of all animals, really possess this ability? In a study published in 2020, New Caledonian crows were first shown one of 3 puzzle boxes that they already knew how to open, each requiring a specific tool. They were then removed from the test area for 5 minutes before being presented with 5 different objects. 1 tool for each of the boxes and 2 distraction objects. After selecting an object and waiting 10 minutes, they were allowed back into the test area. The researchers discovered that not only did the birds choose the correct tool that would open the corresponding puzzle box, but they would remember to bring the tool with them into the test area. In another study, researchers presented the birds with a locked box with a treat in a similar manner. But this time...
Yeah, sure. I think I take your point of, like, whatever, the course of human history, sure. Much longer than 20,000 years. But even then, just because something is doubling over, you know, a shorter time scale, like that is still in the ballpark of evolution. That is totally something that evolution can do, or natural selection can do. You'd have to show that, like, you basically have to show that evolution could not have produced this, and therefore it must be some other explanation. But evolution totally can produce a change like that. While it might seem drastic in comparison to, you know, other things, but that is still, like, that is well within evolution's ballpark and skill set.
The birds were then presented with an assortment of items, including the correct tool, a few distractor tools, and an immediate food reward, but one that was less appealing than the one in the locked box. And most of the time, the birds took the risk and delayed their gratification, kept the correct tool, and later opened the box with the better treat. This might seem easy to us, but it isn't. Only once human children reach 5 years old does this ability arise. And even then, it doesn't in all of them. For some, it takes a few...
Oh, well, I was going to end the stream after this video anyways, but thank you so much for passing by. Super grateful, and thank you so much for all the great questions, and well, pretty much all the great interaction that you brought. Always a pleasure, and have a lovely evening.
Few more years. These mental skills certainly set crows apart from other animals, with their actions and abilities more in line with the smartest of the primates. How does this bird give the most intelligent primates a run for their money? Why does a bird need to be this smart? Even though crows may have a similar intelligence to many primates, bird brains and primate brains are not at all the same. Birds and mammals split on the evolutionary tree 320 million years ago. And since then, their brains have become quite distinct from one another, making scientists question exactly why corvids are so smart. In vertebrates, the pallium is the gray and white matter that covers the brain. Before the evolutionary split, both birds and mammals evolved a large pallium. But after their lineages split, the mammalian pallium evolved into a layered structure known as the cerebral cortex. This is the most complex structure of the mammalian brain. Each layer contains nerve fibers running horizontally and vertically that process and transmit information. Here is where many important functions take place, including learning, memory, sensory perception, and conceptual thinking. Birds don't have a cerebral cortex. But a study published in 2020 found that the fibers in circuitry in the bird pallium are organized very similarly to a mammalian neocortex, with fibers running both horizontally and vertically.
Thank you so much for the $10. Super generous. Well, you've been super generous for, well, ever since you got here. And again, can't thank you enough. Have a lovely evening, and I look forward to the next interaction. And take care and be well.
Vertically. The authors believe this explains where bird intelligence comes from. Another reason why birds were initially thought to be incapable of this level of intelligence was due to the small size of their brains. But it's not the absolute size of an animal's brain that matters, but the brain-to-body ratio. Crows tend to have big brains compared to their body size. In mammals, the bigger-brained individuals are humans and dolphins. In birds, it's parrots and crows. However, as scientists' understanding of the brain grew, they discovered it wasn't just size that mattered, or even brain size relative to body size. A big proponent of an animal's intelligence is the density of neurons. This is because having tightly packed neurons leads to better communication between them. And it turns out that certain bird species have very densely packed neurons. In a 2016 study, researchers measured the number of neurons in various bird and primate species. They found that some birds, like corvids, have twice as many neurons as primates with similar-sized brains, with numbers closer to that of larger primates. While these findings tell us how crows are so smart, the question remains as to why. Plenty of bird species thrive with fewer neurons and weaker mental abilities. So why did crows evolve this way? One theory is that it has to do with the way they are raised. Unlike other species, corvids spend more time with their parents. New Caledonian crows are fed by their parents for up to 2 years. During this time, they have ample access to role models who are making and using tools, and have the opportunity to use these tools themselves. This extended parenting time allows skills to be transmitted vertically, meaning from parent to offspring. The family groups of many species of crow can be very tight, with young crows even helping to defend their parents' new nest of babies. However, beyond tight family groups, adults of many species of corvids don't lead particularly social lives. New Caledonian crows mate for life, but otherwise don't really interact with other adults. Other species of corvid will often forage and roost in large groups, or work together to mob a predator, but there isn't a social hierarchy or complex social bonds in these settings. This seems to go against one leading theory of intelligence, called the social intelligence hypothesis. Chimpanzees, for example, live in large and complex social groups, where they have to interact with others outside their immediate family and keep tabs on many relationships. It's a constant game of politics. Dogs, wolves, and even orcas have to deal with a great amount of social complexity. And this social intelligence needs a lot of brain power. If adults of many of the smartest corvids don't live in complex social groups, why would a similar intelligence have evolved? Adult corvids may rarely live in wide social groups, but often juveniles do. Before pairing up with a mating partner, during their teenage years, some species of corvids live in larger flocks, aptly called juvenile gangs. And these gangs work exactly like they sound. A bunch of teenagers hang out and bully each other and fight. In these angsty years, corvids, like ravens for example, form the many relationships that they have to keep tabs on. And intriguingly, corvids may need social intelligence to interact with other species. Outside of Yellowstone National Park, certain ravens have been seen interacting with wolves, especially wolf pups. The animals have been seen playing tug-of-war, and the ravens tease and entice the puppies to jump and grab sticks. Sometimes the ravens will even pull the wolves' tails. By all accounts, it looks like the animals are playing. Some scientists think that individual ravens may even develop special bonds with individual wolves within a pack. This relationship building may help the ravens more readily scavenge the wolves' kills. This propensity to interact with other species has some groups wondering how we may be able to cooperate with corvids. One idea is to train crows and other corvids to pick up cigarette butts or garbage. A few different companies are developing devices that will autonomously train the birds, giving them a treat once they place the trash into the device. A theme park in France has already been undertaking a similar operation. They trained 6 rooks to pick up trash 4 days a week under the supervision of the park's falconer. While it may seem like we're taking advantage of these birds and their intelligence by having them do something we should already be doing ourselves, those involved seem to have good intentions. It helps the environment, teaches humans to cooperate with another species, and teaches the public about these intelligent creatures. By studying other intelligent species and what made them that way, scientists have gained a better understanding of the brain and its development. And for all we know, we could be overlooking other small-brained species that could harbor a higher intelligence, other animals that can help us unlock the many secrets of the brain. I love talking about animal intelligence on this channel because I love the way the brain works. I love its mysteries and its unbelievable power. And because of this, I love the feeling of learning, of grasping new concepts, of those moments of sudden understanding washing over you. When physics and math and chemistry and biology start to click together, feeling like one beautiful interconnected machine that explains our universe. And I'd like to think that crows feel this too when they get their treat out of the tube. And to engage myself in STEM learning, I use...
Righty, add. Alright. With that, gonna end the stream here, gonna look for someone to raid. I think I'll raid into Squiddideer. But yeah, I want to thank everyone that passed by. Super grateful and appreciative. Anosmia, thank you. Bob Marley, Oily Olive, Noda Bud, and anybody else that I missed. Towler, thank you so much for passing by, and the sub, much appreciated. Always grateful. And yeah, I think the next time that I should be streaming should be on Friday. I will be going on Wick's panel about, you know, identity, essentially. That starts at 6 p.m. Eastern. So I plan to stream that. And then we'll see, maybe we'll get up to something after the panel. And then I should be doing my usual stream on Sunday. Still trying to work out what that'll be. Maybe I'll go back to psychedelic research. I'll continue that series. Covered MDMA. Maybe now I will cover, you know, LSD or psilocybin, something to that effect. We'll find out. But until then, yeah, thank you everyone that passed by. Super grateful. We are going to raid into Squiddideer. They are a fellow lefty-based streamer. Give them a follow, give them support, and until next time, take care and be well.
Enough by just cutting the food into small pieces and then leaving it out in the hot sun. That works. But
no visitors. I'll lose my job.
Oh, boo-hoo. I'll lose my minimum wage job.
Dr.
Corey Campbell.
You're mean, Rutherford B. Hayes.
Listen here, young man. We have nothing to fear but running out of beer.
All right, one small get-together. But let's keep it quiet and dignified.